starting with fungal and moving into bacteria and more. So it's going to be a good day. If you can stay with us the whole day, that's great. If you have to pop off and get back on for the recordings on Canvas later, that's a great way to do it too. So now I'll let Tamla do a little bit of introduction and yep, well, you can get started, Tamla. Thank you so much.
All right, good morning, everyone. My name is Tamla Blunt. I'm a faculty instructor at Colorado State University, which is a new position for me. I was just actually promoted in January, so I'm really excited to be focusing on teaching. And so this is actually part of my, teaching. And I love doing this. I love working with master gardeners. So one of the things that I wanted to kind of caution you about today is I had actually planned on being down in campus or on the CSU campus to deliver this presentation today. Unfortunately, we got 40 inches of snow at my house and I am not plowed out yet. So I was not able to make it down to campus. So I'm working from home and you may notice a little bit of lag time in some of the internet. So once I get started with the presentation, I'm going to take myself off video. I'll still keep my audio on to kind of preserve bandwidth. I have satellite internet up at my house in the mountains. And so we'll be doing that. And then, as we move through today, there will be a Q&A. If you have questions about anything, Mari and Cassie will be monitoring those, and there will be points in the there will be areas in the presentation where I'll stop and, I'll ask Cassie if there have been any questions or anything like that. So other than that, I hope that you enjoy the plant pathology session today. And if you have, like I say, if you have any questions, please put those in the Q&A and we will try to answer those at some point today. Someone raised their hand, but I'm not sure what kind of
We'll ask questions through the Q&A, please.
Cassie, do you? Okay. All right. Thank you. Okay. So I'm going to go ahead and share my screen and we will get started with a plant pathology and talking about that. And here we go. So last week y'all had IPM and plant disease diagnostics. Some of this will be a little bit of an overlap because when I talk about plant diseases and specifically problems of plants, it's really important to make sure that we know what we're dealing with. And so I always frame it in the terms of diagnostics. And so when we start looking at this, this whole diagnostic process and trying to figure things out, it's like, well, why do we need to know about plant pathology, right? And that's because a lot of the questions that you're going to get, whether it's help desk or email or phone calls, they're going to be about plant problems. And I can tell you, even though we just had this humongous snowstorm that came through, in about a month, probably people are going to start getting cabin fever and they're going to be out in their yards and they're going to be out in their landscapes and they're going to start calling master gardeners because they see something. We don't know what this is. We want to know. And so they're going to be calling you. And so a lot of the times it's like troubleshooting what's going on in the landscape. And a lot of the plant problems are, that's just it. So this presentation is designed to give you a background in all types of plant problems. We're going to talk about abiotic versus biotic, and I'll define those as we move through today. We're going to, I've got lots of different terminology in here on different definitions and descriptions. I always liken plant pathology to a foreign language. We all have our own specialties in what we do, and they all have a unique language to them. And so plant pathology is no different. A lot of the terms that we use are, instead of saying round, we say, or what do I want to call it? Instead of clear, we call it hyaline, or, something along those lines. So it's just kind of a little bit of a difference. And so I try to describe those and to define those as we move through the presentation. Also in plant pathology, we use terms like generally, usually, consistent with, sometimes, and it depends. And this is because sometimes it's never an absolute in plant pathology. There are several, lots of different things that can go wrong with plants. And so we say, well, and especially if we don't have a way to properly identify what is going on with a plant, it's just like, well, generally when we see something like this, it could be, or it might be, or something along that line. And hopefully this presentation, this plant pathology, will give you a little bit of a background in helping you with that diagnostic process and determine what questions to ask your clients, whether it's on the help desk or whether you're in the green industry and you have people coming in and say, what's wrong with my plant? And they're holding this up. And then you can start asking them some questions about, the care of the plant, different things like that. How's it being treated? When was it planted? What do the symptoms look like? What do the signs look like? And I'll define both of those as well. And so when we look at this, it's all, all of those things put together. So to start off with, when we look at plant problem diagnosis, well, diagnosis, this is the process for identification of problems. CSI, the television show, hasn't been on television for a while, but we used to say, consider it CSI for plants. However, that's probably for an oldie like me, because CSI is crime scene investigation, and they use lots of different tools to identify the murderer, or figure out who the suspect was. And so that's kind of what we do with plant pathology. We use entomology, which is the study of insects. We use plant pathology. We use plant identification. We use biology of plant, horticulture. All of those things are involved in plant problem diagnosis, because a lot of it starts with, what kind of plant do we have here? And so we have to know what that is in order to be able to understand what's going on. And so basically, it's also a little bit of the, it's a little bit of a investigation, and it's a lot of detective work. And so we look at things like this, and, we try and figure out what's going on. And so when we do this in steps, and we also have our tools with us that we can use. So some of the things that are handy to have if you are working with plants and you're trying to look at different things, a hand lens or a magnifying glass, binoculars even, so that you could see maybe the tops of the trees or what's going on up there. A pocket knife to kind of dig around or to maybe cut branches off. A soil probe or a shovel can also double as a soil probe. A small notebook with pencils is with a pencil is always good to have to take notes. I don't know about you guys, but that's always the biggest lie that I tell myself, is that I'm going to remember that. And no, I don't. I have to write things down. Because otherwise it's like, oh, where did I find that sample, or who brought that in, or what was associated with that plant that I need to know, and things like that. So it's really good to do that. And the only reason I say with a pencil is because if it's, say, an insect sample and you want to keep it in alcohol, you can write on a piece of paper in pencil and stick it inside the alcohol. Stick that paper inside the alcohol, and the pencil will stay, and you'll be able to read your notes later. If you write it in pen, the alcohol will dissolve the ink, and so you won't be able to remember or see what was written on there. Plastic baggies, small saw or hand pruners are always good. And, for if you are a gardener, if you work in the industry, if you are a master gardener, I am sure that you probably have all of these at some point. People look at my car, sometimes I'll be going somewhere and they'll look at my car and they'll see my gardening bag and they'll see, things like that. And they'll go, do you always carry these things with you? And I'm just like, doesn't every gardener carry these things with you? Just because it's like, well, yeah, you never know when you're going to find something really cool that you want to bring home, or you want to look at a little closer and you want to see what's going on. So it's always fun to collect samples. So let's talk about what a plant disease is. And a plant disease is anything that's abnormal on a plant. It could be an abnormal growth. It could be abnormal leaf spots like the picture in the upper left. It could be abnormal fruit like the picture in the middle. It could be sort of an overgrowth like the picture on the right, where you're getting all of these flowers growing together. So it's anything that really it's not normal. And then we are trying to figure out what causes that, right? And so that's when we start looking at potential things that can happen. So when we look at this, we look at the plant disease, the pyramid. Plant disease pyramid, or often called the triangle as well, where we're looking, we have the host plant, and it can be a susceptible host plant. We have the environment, and then we have the causal agent. And those are the three corners of the triangle. Now, if we have a susceptible host, we have the causal agent, whether it is a pathogen or an abiotic issue, and we have the correct environment for that pathogen to grow and to infect the host, then chances are we're going to have a disease problem. Now, if we knock any one of those three corners out, if we don't have a susceptible host, even though we have the right environment and a causal agent, then we're still not going to get a disease because the host is not susceptible to that particular pathogen or causal agent. And so we want to look at these different things and say, okay, and if we don't have the right environment, well, probably not going to get a disease either. So a lot of times all of these three come together and, create the perfect storm, in other words. So as we walk through the plant diagnostics today, or the plant pathology portion of it, we're going to kind of keep in mind the plant problem diagnostic steps, because the most important thing to identifying what could be wrong with the plant is knowing what the plant is supposed to look like and what the plant is. So the identification of the plant, knowing the normal appearance of the plant, is really important. And we'll probably get through symptoms and signs before we get to our first break. So hopefully that will work out really well for us today. So we need to identify our plants. And number one, this is because a lot of our pathogens will affect certain plants. A lot of our plants have certain pathogens that affect them, so they have the known enemies. And then we can also look at the plants and say, well, is this normal? Is this plant supposed to look like this? Or is it normal for this time of the year? For example, if somebody comes in to you and, calls you in at the end of September, or brings a plant in at the end of September, and the leaves are starting to turn yellow and fall off the tree, and they say, well, what's wrong with my tree? And it's like, well, we have to start asking questions like, well, when did this tree start turning yellow? I mean, did it happen in June? Well, then we may have a problem. But if it just started happening at the end of September, that might be normal, because the trees are starting to senesce, to harden off, to start losing their leaves and get ready for winter, right? So again, it just depends on what is happening with the plant and what it is supposed to look like. So for example, in these particular pictures, knowing the normal appearance of the plant is really important. The upper picture in this one are juniper berries on a plant. These are the flowers of the junipers, but I have had people ask me, bring this to me and say, what are these galls on my juniper? And it's like, they're not galls. It's just normal. This is normal for the plant. The lower right picture is a bristlecone pine. And if you are familiar with bristlecones, they are a 5-needle pine. They produce rosin from the needles that dries, and it has this whitish appearance on the needles. So it sometimes looks like scale insects. And so, but it's not. This is absolutely normal for a bristlecone pine. Now, the picture on the lower left this is not normal. This is a ponderosa pine, so it's a 2 or 3 needle pine, and it has whitish scales on it as well, but that's an actual insect. So again, knowing what's supposed to be normal versus what's not. So the bristlecone pine is normal, but the ponderosa is not. Another thing that's really important is knowing the scientific versus the common name. If you're in different parts of the country, people have different common names for the same plant. And so sometimes people will move into our area and they're calling a plant by a common name. They don't know the scientific name, and we're not familiar with that. And so we have to do some research to figure out what it is. Knowing the normal, and it can be really important to know that. Again, knowing what's normal. I once had a call from a forester in Southeast Colorado, and some Coast had moved into the area, and they a hedge of juniper. And so these people were calling because yellow on top and junipers, and they were not greening up, and they weren't, knowing that it wasn't changing the plant, right? And so I asked if they could send me some pictures. And so they did. And I looked at the pictures and I was like, I know what this is. And what it was I suggested to her that this was probably a cultivar of juniper called Old Gold that has the yellow tops and the green is down below, but the tops will never yellow because that's the cultivar. That is what it is supposed to look like. And so once I said that, she's like, oh, yeah, I bet you're so right. And so she went back to the homeowner and they figured it out. And so she was able to talk with them. But again, this is knowing some of these varieties and some of these cultivars and what they're supposed to look like. And that's important. So in this case, for this plant, this picture on the right, is this normal for this plant? And the answer is yes, if you know that it's a corkscrew willow. Otherwise, if you were to look at this, you might say, well, looks like maybe somebody sprayed herbicides on it or something, but it's not. This is absolutely normal for this particular plant. It's a corkscrew willow, so it is supposed to have this sort of, curled petioles and curled stems and things like that. Then we look at this plant in the bottom left, and we're like, is this normal for the plant? Again, absolutely. This is Harry Lauder's walking stick. And so the branches are very twisty and curly. And so consequently, it's very beautiful plant. And I love these. I love the texture of the plant and how it looks in the landscape, and it's really fun. But as I mentioned, I live in the mountains, and I planted 2 of these in my landscape, and the deer ate them. So I do not put plants out there for the deer to eat. So unfortunately, but anyway, that was normal. So once we know what plant we're working with, we've identified it. We know what we're dealing with. We know what it is supposed to look like. We can look at symptoms and signs. So symptoms are the plant is telling you something is wrong with it, and you're not quite sure what is happening. And so when we look at these things, we see all sorts of different symptoms. So we can see yellowing of the plant or chlorosis, which is loss of green color. So the leaves might be turning yellow. So what could be causing that? And we'll talk about some of the causes of chlorosis a little bit. Chlorosis by itself is not a very definitive symptom, excuse me. There are lots of reasons that a plant could turn yellow. It could be the time of year, could be overwatering, could be underwatering, it could be nutrient issues, different things like that. So again, it's not totally cut and dried as to what is happening. So some of the symptoms that we could see are mosaics, where we see these alternating yellow and green or white and green on plant tissue. In this case, this picture is a rose mosaic virus, and this is caused by two viruses, prunus necrotic ring spot and apple mosaic virus. And so those two viruses together can cause rose mosaic virus. And we'll talk about viruses as we get this afternoon when we get into the specific pathogens. But just to kind of give you an idea, it's going to depend on what type of rose you have and how severe the infection is and things like that as to what kind of patterns, because it may not always present just like this. We can also see necrosis or dead plant tissue. So necrosis is dead plant tissue. We can see necrotic spots here. So we have necrosis in the spots in the lower right picture. This is actually Marsanina leaf spot on aspen. On the left-hand picture, we see this necrosis. This is also called a canker, but this is dead plant tissue on the trunk of a tree. And in this case, this one is caused by Cytospora. In the upper right picture, we have the tips of the leaves are dying back on this one. And so we could see that, yeah, something's happening up there and we'll need to figure it out. So this is necrotic tissue on that upper right picture that's starting in the back. We also have what's called blight. We usually associate this with sudden death. And so when we talk about it, it could be the tips of the trees are dying back, so sudden death. So fire blight in this case is what this picture is. And so we have the blossom blight, the pathogen is moving back into the stem and it's causing the leaves to die back. And the stem to die back as well. And so we call it blight because it looks like it happens very suddenly. We also have wilting, which is limp or droopy plants where the leaves are drooping. They're also, let's see, there's flagging is another term for wilting, where the branches of a tree could just be kind of drooping down or leaning down. It looks like it's flagging a little bit. And this could be caused by, overwatering, underwatering, could be a pathogen, root-borne pathogen in there. So again, all sorts of different problems that could be causing the wilting in the plant. And so we want to make sure that we're, doing our due diligence in proper diagnostics. And again, canker, like I mentioned before, is usually a necrotic area. It could just be discolored. It could be sunken. It may not be sunken. It just depends. And it's usually on the trunk of a tree. So these are things to kind of look for when you are looking at plants. And sometimes you may not be able to see this whole plant. You may just get a picture, or you may just get a description. Somebody might be telling you what this is supposed to look like. In that case, it's often good to get a picture and say, oh yeah, that's called a canker. And then we can figure out what's causing it based on the type of tree it is. And if you look up close, you can oftentimes see fruiting structures of the particular pathogen. In this case right here, in the upper part of this picture, you see these little pimple-like structures, or these little mini volcanoes, if you will. And these are the fruiting structure called a pycnidia of Cytospora canker. And you can see where the canker is moving. You can see the margin edge of the canker down here in the lower portion of the picture, where you have that distinction between the light and dark tissue. And so you could see, oh yeah, that looks like a canker in it, and it's moving within the tree. We can also have stunting of plants. This is stunting, of course, means the plant's not growing very well. It's smaller than it should be. It's shorter than it should be. And so we're trying to figure out, yeah, what's going on with that. And so we are, trying to get with this. In this particular case, this picture is from a greenhouse study at CSU, and it was to deal with nutrients in corn plants. And you can see the different coloration in some of these plants and the height of the plant based on whether they were being fed the correct nutrients or not. So you would be able to see something that looks like this. Sometimes we might see swellings on the plants. So these things are what we call galls. In this particular case, this is Western gall rust on ponderosa pine, I believe. And so this is a fungal pathogen causing this gall. Now, sometimes it could be an insect causing galls. It could be pathogens causing galls. It could be an abiotic issue. Maybe some girdling of the tree is causing gall-like growth on the plant itself as well. So again, we have to figure out, okay, what's causing this particular symptom. So again, the symptom is the plant is showing us something and we have to figure out what it is. We also have distortion. So again, something is deforming on the plant. It could be the leaves, it could be the stems, it could be the petioles, it could be twisting and curling of the plant material, all sorts of things like that. So again, you know, we're trying to figure out what's going on. The left-hand picture here, it looks like an overgrowth, a gall overgrowth of some sort. And I honestly can't even identify that plant. I would have to look a lot closer to be able to see that. The picture on the right-hand side is powdery mildew. It's a bad case of powdery mildew, but it's so bad that it's causing the leaves to curl up and to distort. So again, you know, so we see that distortion and then we have to try and figure out what's happening with that particular plant. And then we also look at where are the symptoms? Are they on the leaves? Are they on the or on the roots? Or is it on the stems? Is it on branches and things like that? So we look at this, and this particular photo is Phyton 27 damage to poinsettia. Well, it's Phyton 27 is a pesticide that is used to control insects and diseases in the greenhouse. And apparently this particular cultivar was very sensitive to that treatment. And so it started turning yellow. So again, it caused some what we call phytotoxicity issues with the plant itself. So, you know, we look at the leaves, then we look and see, is it on the stems? On this particular picture, you can see the canker on the stems. You can see that distinct difference between the brown and the green tissue. And so the brown and the green tissue here, you can see what's happening. You can see that something's happening, and now we have to figure out what this is. And this is actually a poinsettia. And you can also see that the leaves are wilting on this particular plant in this picture. And so these stem and leaf symptoms can be expressed, but it may be the roots are having the problem. And so sometimes we have to look at the whole entire plant in order to figure out what's going on. And in this case, this is what it looked like, this picture on the left here. And then if you pull it out of the pot, you may see something that looks like this. The roots are brown and mushy. And so there's an indication that that's a root rot. Now, which pathogen is causing this root rot? We don't know just by looking at this picture. We would have to do some microscopic slides to look and see if we could see fungal structures, or we would have to do artificial culturing at a diagnostic clinic, things like that. And so again, you know, looking at something like this. Now, this picture shows you the roots that are on the left-hand side of this picture are nice and tan or light tan and creamy color. That's an indication of healthy roots. When you see the dark brown, mushy type roots in the middle of in the picture here, that's an indication of root rot, that something's going wrong with the plant. And oftentimes with root rots, you won't see that until you start seeing the above ground symptoms of the wilting of the leaves or the yellowing of the leaves, just because there's no water or nutrients getting to the top part of the plant because the roots are rotted and they're not able to take up any water or nutrients. And so consequently, all we see are the upper parts, upper symptoms, but we're not seeing the root rot yet. And so this is that particular plant. It was a grass that was being grown. I don't think this is corn, but again, you can see they pulled it out of the pot and it had this massive root rot problem associated with it. We also look at the symptoms at whether they're on flowers or fruit or as the picture in the right on the flowers as well. So on the fruit on the left, this is plum pockets. You can see that it's damaged, and that is a fungal disease associated with some prunus species. The picture in the middle is petunias with botrytis blight. And that again is a fungal disease, but you don't know that because all you're seeing are the symptoms. You wouldn't know that until you start looking at the plant a little closer, doing a microscope slide or sending it off for diagnostics or things like that, just to make sure what it is. But that upper picture with the petunias and the holes in the flowers and things that look like that, and it looks like the edges are eaten, that could also be geranium budworm, an insect that loves petunias. The larval stages love to feed on the flowers, and so consequently they leave holes in there. So again, these are some of the things that we think about when we're looking at symptoms. It's like, okay, what could cause this? What do we need to look for? And that can help narrow down what you're looking for, because again, your host, not everything, not all pathogens and all insects affect every single plant. They're usually somewhat host specific, and so consequently you have that that ability to kind of narrow it down when we're looking at the cause. So after we've looked at the symptoms, and remember the symptoms is the plant reaction to whatever is happening to it, then we start looking at signs. And this is the presence, or we see something, we see the actual organism that's causing the problem. And probably the best example of this, is things like fire blight, where we're seeing these ooze coming out of the fruit, or there's ooze coming out of the trunk because it's a bacteria and it's moving through the vascular system of the plant. And so it can come out of the lenticels or, the stomates, different things like that on the plant. And so we're seeing these things. Another thing that we can look for are some of the fruiting structures of fungal diseases. Things like Cleistothecia, which is the overwintering structure for powdery mildew, and it can be on plant material that's in the landscape. Right now it's buried under snow, but you might be able to see it at some point. The picture on the right is a structure called anacervuli. This is anthracnose. So you might see this on, say, like ash leaves or oak can all get anthracnose as well. And then there's another structure on the bottom picture that's called a parathecia. It looks kind of like a little vase. And so you would be able to see those structures coming out, erupting out of the plant material itself. Cleistothecia actually look like somebody sprinkled pepper on the leaves. And so once you look at it with a hand lens, you would be able to see that they were the structures and stuck on the leaves, basically. You might see fruiting structures that are embedded in the plant tissues. So for example, this is an anthracnose pathogen as well. And the arrow is pointing to the acervuli, and you would have to come up really close with a hand lens to be able to see the setae that are associated with that. You can also see hyphae or mycelium growing, and this is an evidence of a fungal pathogen. And so the picture on the left is a carrot with mycelium growing all over it. This is actually Sclerotinia sclerotiorum. It's a fungal pathogen. It affects beans and carrots and different things in the vegetable garden. And it also produces a black structure called a sclerotia, which you can see kind of right here in the middle of this picture, these dark black structures right here. These are sclerotia. And those can survive in the soil for a number of years, just depending on environment and host plant, whether they will start growing more mycelium and infect the plant again. The picture on the right is a picture of Dutch elm disease growing in culture. And it looks very pretty. It's interesting from a plant pathologist standpoint. From a landscape type standpoint, it's not a good pathogen to have because you have to cut down the tree. But again, you would see structures that look like this. And so you would see all of these little strands right here that are growing off of it, that is the hyphae of Dutch elm disease. And then the hyphae is actually an individual thread, but the mycelium is all of the hyphae growing together. And so when we see these, things like that, we'd say, oh, that's a fungus, and then we can try to identify it. In this picture with the Dutch elm disease, we can also see these are a structure called synema. This is what is produced by Dutch elm disease. And as this is kind of a gross description, but as I tell my students, it looks like a little black toothpick with a ball of snot on it. And this little ball right here will contain millions of spores of Dutch elm disease that can be moved around by beetles and move through the vascular system of the plant. So it's really kind of disgusting, but again, it's descriptive and the students don't forget it. So we can also see spore structures. This is a chrysanthemum rust on a chrysanthemum plant, and these are on the underside of the leaves. We see these orangish rust colored masses, and that's pretty much why it's called a rust. It's a rust disease, and that's the name of the pathogen. And so there are different types of rust pathogens that we have, and each of these orange pustules here, they look like little pimples, and then the spores inside actually look a lot like pollen, but they're really not, but they're fungal spores, and they can get blown about by the wind and spread to different types of plants. Especially in a greenhouse, if you're growing other chrysanthemums, they'll get spread over to those, because rust diseases are host specific. So these won't go to like geraniums. That's a whole different rust. So rust pathogen. So we also have mildew, powdery mildew, which is the whitish growth produced by specific fungi. As you can see on this rose, you can see all this white color here. This is all powdery mildew. And powdery mildew is actually both a symptom and a sign, because it's showing on the leaves, and then when you get up close to it, you can actually see the fungus growing on the leaf surface, and it grows on both the upper and lower leaf surfaces. And so it spreads by wind, because or splashing water can move the spores to other leaves, and so that's how it moves. Powdery mildew is also mostly host specific as well. And so it will affect rust disease, or it will affect, like rose powdery mildew is probably, it's not the same, the one that goes to your lilac and things like that. So you have to, those are two different powdery mildew fungi. But again, you start seeing that whitish growth, and okay, powdery mildew, yeah, it's a fungus. And so we see that, and it's a sign. We can also look at mushrooms, otherwise mushrooms growing, say, like from a compost pile. Maybe somebody has, put a lot of compost in their garden, things like that, and it's growing mushrooms. And so mushrooms are the fruiting structures of a group of pathogens called basidiomycetes. And these guys, some of them are edible. We eat them, when you get them from the grocery store. Those are the edible ones. I do not, unless you are a mycologist and you know definitively how to identify these, I do not recommend foraging for mushrooms, just because unless you can identify them. There's a lot that don't taste good. They're poisonous, and you just really don't want to eat them. When they're growing out the side of a tree, like this picture on the left, we call it a conch. And so it's a fungus growing out. And a lot of times, these particular mushrooms and conchs are associated with wood decay pathogens, which we don't often see in the landscape. But up in the mountains, I see them fairly frequently up here. I could just walk around and, oh yeah, that's got decay. I see that now. And sometimes they don't produce conchs. You don't know that a tree is decayed, but sometimes they do, and you could see those. I know that I one time got some of that mushroom compost to use in my raised beds, and I put that in there, and I think I had mushrooms coming up for 2 years. I did not eat them, but it was kind of annoying. So symptom versus sign. We have the symptom is the effect, basically. So we're seeing wilting, or we're seeing some sort of canker down here towards the crown of the plant. We're seeing root rots associated with that. So those are the effects, and that is the symptom. The sign is actually going to be the cause. So in this case, we're looking at the sign where this is the mycelium that is coming off of these roots. And in this case, I know you, it's hard to tell, but this is most likely rhizoctonia. The hyphae is quite large, and it has this right angle branching associated with it. So it's, the sign is the cause. And so when we start looking at that, then we try and figure out what it is. So symptoms versus signs. So for example, with a lilac, if you were to look at a leaf that looks like this, you can see the mildew on these leaves here. Turn them over and you can see the powdery mildew on the underside of the leaf. And you can also see these little areas in this bottom picture. You can see these little dark areas. And if you look up close, these are the cleistothecia. And these start forming when the plants start senescing for the winter. And so as the plants start senescing, the powdery mildew, of course, because it needs that live host tissue, and our plants the leaves don't stay over the winter time, they form these structures so that they can survive the winter, and then they will infect the host in the next spring. Once the temperature warms up, we get a little bit of moisture, the plants start leafing out, and these structures called cleistothecia will open up and eject spores that can land on the leaves and start the infection cycle all over again. So one of the things that you will hear me say quite frequently as we move through this presentation today is sanitation in the landscape is important, simply because if you remove a lot of this leaf tissue, your chances, you've reduced your chance of reinfecting the plants by, tons. I mean, if you get rid of all of the leaves. Now, that's not to say that you will not ever get powdery mildew again, but hopefully it will be a lot less than what it was the previous year. And so you'll be able to see that. So again, this is just an example of, these are hawthorn leaves. And on the upper side of the leaf surface, you can see these yellowish areas right here and you can see these orangish yellowish structures or spots on the leaves right here. But if you turn them over, you see these little things that look, kind of like little miniature flowers, or some people might liken them to spiders with lots of extra legs on them or something like that. These are the fruiting structures of pathogen called gymnosporangium rust. And this affects junipers and hawthorns or apples. And it has two alternate hosts. And we'll talk about that as we move through today as well. But you can oftentimes see some of these things associated with it. So you would be able to say, oh yeah, I know this is juniper hawthorn rust, right? And if you look up close again, you can see here's those fruiting structures. These are actually called the scientific name for these is Esia, and it is the esial stage of the gymnosporangium rust on the hawthorn. All right, are there any questions about symptoms and signs and overall view of plant pathology? Cassie or Mari, do you guys have any questions for me?
I think one question is clarification on sterilizing tools. We had that come in a couple of different times.
I'm sorry?
A question on sterilizing tools. We had that come in a couple of times.
Is it in the question box?
You might have to dig a little bit. Just clarifying on how the process of sterilizing your tools.
Oh, sterilizing your tools? Oh,
yes.
Okay. I haven't gotten to that part yet. We'll talk about that when we get to fire blight this afternoon in pathogens. And so we'll talk about sterilizing tools then.
Perfect.
If they can hold on. Okay.
Yep. Were there any others you were seeing, Mari?
Oh.
Yes, there is one that is, do you have any recommendations for a good key to diagnose certain plant diseases?
A good key?
Yeah, I assume like.
Sorry, I don't mean to laugh. I don't mean to laugh, because it depends on the plants, and it depends on what's wrong with them. Probably, there's not one definitive key that I can point you to. I know that the insects and diseases of Colorado that Whitney published a few years ago is probably the best one for our area because it covers a lot of what we have here. And so you may be able to find that, may be able to find it in there. However, the key to that is still you need to know what plant you're looking at. And so a tree key is really good to, or, a plant identification key to identify the plant can be helpful as well. So yeah, Whitney's book is probably the best one that, for getting you started, and you may have to go to other sources as well. There's lots of different references that you can use, but one definitive key for everything, nobody's written that one yet.
I think that's it. You're good to go.
Okay, well, next we get into abiotics, and it is 9:52. Do we want to take our break a little bit early?
Technically we have until 10:30, so it's up to you.
Oh, okay. All right. Well, we'll keep going. 10:30 for the break? I thought it was
Yep.
10 to 10:15.
No,
Okay.
we're till 10:30.
All righty. Okay, sounds good. Thank you, Cassie. All right, so let's talk about abiotic versus biotic. So abiotic are what we consider non-living causes of plant problems, such as the environment. Sorry about that. And so these would be things like temperature, snow, nutrients, and different things like that. And so this is what we're going to focus on through the rest of the morning. The biotic problems, these are the living organisms, and these are our fungi, bacteria, viruses, phytoplasmas, nematodes, and we'll talk about those in the afternoon. So this morning, we're going to focus on abiotic causes, such as these are the non-living causes of what's going on in the environment. So abiotic causes, too low, too high temperatures, too much, too little soil moisture, too much, too little light, nutrient deficiencies, soil type, pH, pesticide toxicity, which we'll just briefly touch on. But again, a lot of these things are what we call cultural issues, as far as some of these things we can control, some we cannot. The too low or too high temperatures, we're outside. If we're gardening outside, we have a landscape outside, we don't have any control over Mother Nature. In a greenhouse, we do, but not everybody has a greenhouse, right? Too much or too little soil moisture, we can usually manage that simply because we can either bring out the hose to water, or if we get too much rain, we can hopefully drain it away from our plants if it's looks like it's waterlogging. And it doesn't even have to stay around the root zone for very long with too much moisture in order to cause some damage to the plants. Lack of or excess light is usually associated with greenhouses. So I don't really talk about it too much here. But, occasionally if you're planting plants, make sure that you know what their light needs are. If you're planting a shade-loving plant in bright sunlight, it's probably not going to do very well, and vice versa. So you have to make sure that you're doing that. Nutrient deficiencies we'll touch on again, as well as soil type and pH, and go from there. So weather-related problems. Here we go. Yay. Yearly dry fall winter period. I do not know if the snow is going to bring us out of drought, but we are currently in a drought here in Colorado. It is pretty bad. We are way below our moisture level for the year. It has been very dry this winter. I'm hoping, for all of my complaining about not being plowed out of my driveway, I'm not going to complain because the moisture is good. We need this going into spring. It for me, it's good fire insurance. To the more snow we get, the better off I am. However, a lot of these weather-related problems can cause problems with our plants. They can break branches, they leave open wounds that can encourage pathogens to get in there, or, open wounds that pathogens can get into them, things like that. Drought can be a huge problem. We've seen this before in Colorado. Late spring freezes, prolonged cool, wet weather. We've seen that like in, I'm not sure if it was last year or the year before, but with vegetable gardens, it just didn't warm up very fast, and people were planting tomatoes outside and peppers, and they just kind of sat there. Nobody got a really good tomato harvest last year. It just went from cool, wet to hot, and so that's not very conducive to growing good tomatoes. So we can have early fall snow, which can also break branches because the trees have not lost their leaves yet. Sudden fall freeze, things like the polar vortex in 2013 that came through. That was very hard on our landscapes when that thing came in. Hail, flooding, all of those things can be problems for our plants. We just have to kind of keep an eye on it and see what's happening. There's all sorts of things that weather can do. So high temperature affects plants at high temperatures are generally injured faster and to a greater extent when those temperatures become higher than maximum for growth. And we see this not so much in the like grapes that I have here, or the ornamentals, but we see this in our vegetable gardens. When it gets too hot, our tomatoes don't set fruit because the flowers abort. They just don't pollinate. And the temperatures are too high, so they're above that maximum. We also can see sun scald on our plants. We can see canker formation from that sun scald. We can see leaf scorch, where the leaves, the margin edges of the leaves turn brown, and it's a pretty even margin edge, pretty even browning of the entire leaf. That's leaf scorch. You can see this a lot. You can see leaf scorch a lot in the median strips on certain streets in Fort Collins. You can see during the late summertime, a lot of these trees have the leaves, the edges of the leaves have all turned brown because they get that, blast of heat from the asphalt, and they're just not getting enough water to compensate for that heat in the plant. We also see sun scald, also known as Southwest disease of trees. This is what can happen during the winter time if we do not protect our young, newly planted trees that have young bark on them from that exposure to the UV light. And basically what happens is, it gets cold at night, but during the day, it may not get very cold, but the UV radiation can warm up that bark to the point where you get that freeze thaw cycle. And then you get that bark to basically split, and it can cause problems. The upper right picture, that tree had sun scald, and it actually survived until last year. And now with the whole Shepherdson remodel at CSU, that tree got cut down. And I've got to find my pictures, because I did take some pictures before they cut it down to show you that that tree was still alive. Even with that huge sun scald canker in that upper right picture, it was still surviving. It was doing well. Down here, this lower picture, you can see this a maple, and you can see the sun scald with the damage here. So basically what we recommend is you can use a water-based white paint to reflect the UV off of the tree. You can lean boards up against these trees to help protect it from that UV light. And you can also do paper wraps. You can wrap the trunk in paper, those tree wraps. Usually we say Thanksgiving to Easter is a good time frame, because that protects them in the winter time, and then you don't have that wrap around it all year long, which can actually encourage insects to nest under there, and pathogens might get in there as well, and that bark will get really soft and susceptible to certain pathogens, so you don't want to keep that in there. Low temperature effects actually do more damage to plants than the high temperatures. Low temperature, the worst times to get low temperatures are early fall frost freezes and late spring fall frost freezes. And that's because in the early fall, the trees have not gone into dormancy yet. They haven't developed their antifreeze, if you will, their winter hardiness. And so we get that early fall frost freeze, and it actually can do more damage at that point. Late spring can do the same thing because our trees are coming out of winter dormancy into full growth period, and the vascular system is moving, the trees are starting to leaf out, and then all of a sudden when they get this frost freeze, and it will kill the buds, kill the new leaves that are coming out, and it will cause problems. So cause problems on the plants. So those are the two worst times. Now we can also get cold damage that can cause some root damage or bark splitting, canker development. The root damage could be because the roots are frozen in the soil. The soil just gets cold and it freezes the roots. So this is what spring freeze injury looks like on some deciduous trees. You can see in the upper right picture, this is an ash tree, and it has killed the new growth that has come out in the spring. But you can see that right behind it, there are some green buds that are starting to come back out. So most trees have secondary buds, or axillary buds, if you will, that allow the plant to re-leaf out. Takes a bit of energy on the part of the plant, so we need to make sure that we're watering properly, that if it needs nutrients, that we're feeding it correctly. But chances are it will leaf out. And so again, it looks like that. Now the thing of it is fire blight looks a lot like this damage here in that upper right picture, but fire blight does not affect ash trees. So you have to make sure that you identify the plant and so that you identify the correct problem. The lower picture is also a spring freeze injury on plants. And this is actually an apple tree, but it is not fire blight. It is the spring freeze that's frosted these newer leaves that have come out, and it's frosted the blossoms that came out first. So again, we're looking at symptoms, but we have to also look at timing. When did this happen on our plants? Freeze injury, if we're looking at leaf tissue, sometimes we can tell it's freeze injury just because we kind of get this heart-shaped look to it. In this bottom picture, you can see there's a little bit of necrosis at the very tip. But what happened with that is that the cold actually damaged that growing tip right there. But that's it. And then the rest of the leaf keeps growing around that. So it gives that heart-shaped appearance. And this up in the upper right-hand picture is an aspen leaf. And you can see this is frost damage as well, just causing this particular problem associated with freeze injury. So again, looking at the timing of the damage and what's happening with the plant is important. So on spruce and fir and pine trees, we can look at spring freeze injury, and we'll see the new candles that are starting to come out. Some of them may be damaged by that spring freeze injury, but you can see there are other candles that are not damaged in this upper left-hand picture. And so consequently, we see, okay, it was a spot sort of freeze thing. And the picture in the lower right here is actually the same thing. You can see damage from freeze, but you also see new candles coming out. The newer growth is lighter colored, and then we see that brown tissue, but the growth here is you can see this in the background. It's blurry in the background, but you can still see that lighter green color is coming out, and you can see that as well. So this is on rose flowers. This happened in 2008. The roses, as these flowers were developing and the floral organs inside were developing, there was a cold incident. And so when the flowers actually opened up and bloomed, it causes this damage called bullheading, because these flowers are not completely formed like they should be, because those organs were damaged by cold temperatures. So a summary of freezes is basically we're going to see damage that shows up 1 to 2 days after the event, generally. And I say that simply because when we had that polar vortex come through in 2013, we were seeing damage for almost 2 years after that, as a result of that frost, as well as a result of that freeze damage to the plants. So again, it's important to make sure that we keep an eye on our, temperature environment and see what's happening. You'll generally see rapid necrosis of, the tips of the plants, the leaves, things like that. You also may see some uniform damage to the same aged leaves, maybe malformation to less severely damaged plant parts. So you could see some curling or something like that. And again, late spring, early fall are the most common times for the most critical low temperature injury for both flowers and foliage. And that's because our plants are still, thinking that it's not time to go dormant or it's time to come out of dormancy. And so we have the vascular system is moving inside the plant. And so water freezes, disrupting those cell membranes. And, a lot of these symptoms can look like foliage or shoot diseases like fire blight, or there's one on aspen that is a shoot blight on aspen. And so again, it's the timing of what we see and when we see it is how we can start identifying that. Moisture problems, low soil moisture effects. Again, when we see low soil moisture or margin dieback or heat stress, scorched leaves on plants. This is in the upper right picture. You can see it's a pretty even distribution all around these little leaflets on this particular leaf. And so you can see all of this is pretty evenly distributed along the plant. And so this is what we would call scorched leaves, because this is usually associated with too much heat and not enough moisture associated with that. It's kind of hard to explain, but it's not a disease. It's it's an abiotic disease, but it's not a biotic disease. It's something's happening with the plant. It's in the wrong place. It's having problems. And so these are things that we need to look at. Again, in the lower picture, you can see the scorch associated with the leaves here. Again, a lot of leaf dieback, but it's very evenly distributed along the leaves as well. So scorch, again, the leaves turn brown from the outer edges and it goes towards the inner part of the plant, usually evenly distributed around the leaf margin. And these oak leaves over here were really stressed out. This is leaf scorch associated on an oak plant, an oak tree. So again, it can be pretty bad. It can be kind of mild. I mean, the leaf on the right here is still able to photosynthesize a little bit, but again, it's something's going on. It needs some moisture. It's probably, maybe in an area where it's getting residual heat. And so it's causing some of that scorch damage. So moisture can also be a problem in the wintertime. It's pretty common on conifers to have winter desiccation. And that's because it's just, the wind comes along, it sucks all the moisture out of the needles, and it causes huge problems. And we, our soils can be frozen. So, and if the soils are frozen, we can't do winter watering. If they're not frozen, we could do winter watering, but oftentimes people don't do it as often as they should. And so we see damage associated with just, needle scorching and twig dieback. One of the things that we can look at for winter desiccation is we're looking at the whole whorl of this needle, this candle right here. And you can see that the dieback is about 1 inch long all the way around these needles. And that's a pretty good indication, and it's got a definite margin edge. This is a pretty good indication that this is moisture damage, that it's not a pathogen. If it were a needle cast disease, it would be more sporadic. It would be interspersed, and it would be sometimes the tip, sometimes the whole needle, different things like that if it were a needle disease. So again, we're looking at all of these things associated with it, with winter desiccation. And you can see, a lot of times we could say with abiotic problems that it's pretty evenly distributed over the plant material. And you can see that in this case, that this is all needle, just moisture desiccation. So some other drought injury examples, again, you can see on these needles, you have this definite band right here. This is associated with moisture desiccation. Spruces will kind of turn purplish color. The needles will turn purpley. And so you could see that that's winter desiccation on a spruce. In pines, they're they're a brownish color. And again, some of the lower needle may turn a little bit yellowish, but it's, mostly you'll see this distinct banding associated with winter desiccation or drought injury. So how does drought happen? Well, it could be because the water is lost faster than the plant can take it up. So it's not getting enough water to replace what it's losing. So a lot of times that happens with our windstorms. There may not be enough water for the roots. The plant may be in the wrong place, and the water could be available, but the roots are not functioning because the soil is frozen. And you may also see like stem or trunk damage that is not allowing the water to get up to the top of the plant. And so we see things that look like this, where we have the crown damage down here, this trunk damage, and it's in a place where it's probably not growing very healthy. The roots do not stay in this little concrete box. They're all underneath the sidewalk, and it's the only way that this tree is getting water is through this little box, which is not where the majority of the feeder roots are. And so consequently, the tree's probably losing more water than it can take up because it just can't take it up. Other things that you can look for on winter desiccation: new growth will usually mask the damage from winter desiccation. You can always feel the buds on your pine trees and also on your deciduous trees. If they're soft to the touch and they squish a little bit, then they're probably healthy. And that new growth is going to mask any winter desiccation out. Truly, a large percentage of the plant has to be damaged before the plant health is endangered. Now, that being said, there are times when something happens and it can happen like that. But other times it's just, for a plant pathologist, I'm willing to put up with a lot more aesthetic damage to plants than maybe some other people. So I just look at it and say, oh yeah, it's going to be fine. And, maybe water it a little bit more or something like that. But again, it's sometimes we have to convince our clients that, yeah, the plant's going to be healthy. It just needs some time to recover. And you don't need to take it out. Now, this is where I could tell you that if you are looking for needle cast disease, on needle diseases on pines, you can see that in this particular picture in the left-hand side, the upper left here, in this circle, this is where you have fruiting structures of disease organisms, and we don't know which one this is, that are erupting through the needles. The cold damage, it starts at the tips and moves down. And we also see this margin edge, and we also do not see any disease organisms coming out of the needles itself. Now, that being said, sometimes on our needles, if we're looking with a hand lens, we can see sooty mold on pine needles. And that is just aerial fungi that is colonizing the needles. It's not doing any damage. And one way to tell the difference between sooty mold and a diseased organism is that with sooty mold, you can scrape it off the needles with, a little, a little pick or something like that. And it's not going to damage the needles. It doesn't pull anything out of the needle. Nothing happens. It just comes off. With a disease organism, if you scrape it, you're going to scrape into that needle or into that fruiting structure of the pathogen. And so you would be able to put that under a microscope and look for spores of the fungal pathogen and see what it is. So how do we manage drought? Well, identify what's causing it. Sometimes it's Mother Nature and we have to add additional water. That being said, if we go into spring and summertime in a drought situation, even this year, we may find ourselves under water restrictions depending on where we live and how bad the drought is. And so we may not be able to water as much as we want to. So we have to kind of balance what we're doing with, water that we can apply to our lawns and trees and things like that, and our vegetable gardens with restrictions that have been placed on us. Fall and winter watering can be done. You can recommend watering once a month to trees and shrubs from like November 1st to March 1st. Fall watering is the most critical time. And this is because the roots are still functioning until soil temperature drops below 40 degrees. So if the soil temperature is above 40 degrees and your air temperature is above 40, that's a good time to winter water. You could do that like once a month. Roots are still functioning, but very, very slowly during the winter time. And so when we water in the fall, it actually helps our trees and shrubs leaf out in the spring because they've been able to bring up a little water. They've actually been able to maybe store a little bit more energy, and that helps them leaf out in the spring. So why do roots not function well? Well, if we are putting plants into areas that have bad soil compaction, maybe they've had construction damage, could be dehydration, they're not getting enough water, could be suffocation, they're too much water, could be planted too deep. So again, all of these things could happen, and it could be why the roots of the plants are not functioning and they're not taking up enough water. Soil compaction and construction damage are probably really high on the list. Planting too deep is probably another issue. And so I believe there's a whole section for the Green School on how to plant. And so I highly recommend that one because planting trees correctly is really important, making sure that they don't have girdled roots, making sure that they're getting enough water, that they're able to sustain themselves. Sometimes it could be oxygen starvation that could be caused by too much water, and the roots are suffocating. So they're not getting any oxygen. There still has to be a good balance of oxygen in the soil for those plants to be able to utilize. If you take that oxygen away, the plants are going to suffer and they're eventually going to die. Usually we see that symptoms associated with this are going to be leaves that are smaller than normal, poor plant growth, yellowing can happen, and again, just a slow death by suffocation. And this can happen in soils that maybe are not ideal, like bentonite clay or things like that, where we're watering too much, but the soils aren't draining very well. And so we have to make sure that we are either amending the soil or doing things that need to be done. Now, that being said, not everything should be amended, but, check your soil profile and make sure that you're doing the correct thing to put your plants in there. And because we want to help our plants grow, right? We don't want to put plants in and just watch them die. We want to make sure that they're going to survive. Soil oxygen starvation, again, too much water, poor soil drainage, not putting the right plant in the right place, too much mulch can be a problem, stem girdling roots or girdling roots, soil compaction. If you see in this upper right picture, if you see moss or algae growing on the soil surface, I'm going to guess that's a little too much water. because that means that the algae has a place to grow, and so does the moss. Soil compaction caused by construction. Soil pores should be, the structure of the soil should be enough so that you get plenty of water and oxygen. So water is the blue line in this picture, oxygen is the white line. And so your soil structure should look something like this, where you're getting good soil, good oxygen mix. If you have soil that looks like this, that's very compacted, your water and oxygen getting into the soil is going to be minimal. And it's going to be very, very slow. If we have too much mulch, or mulch volcanoes, if you will, you can actually, get too much water in there, and it's going to cause a problem because you don't know what's under there. And then the water can hold in that mulch and rot the base of the tree, basically. Also, you can get rodents and stuff moving into that mulch volcano to make a home. It's easy for them to get into. So some of the things that we can see with oxygen starvation are lower portions of the plants. The leaves are dying back in pine trees. You could see the lower branches starting to decline before it gets up to the top of the plant. And so again, making sure that we are putting our plants in the right place, that we're properly caring for them, maybe even check the soil before watering to make sure that the plants actually need that water. Sometimes we just water because, oh, it's hot and it's summer. Well, maybe the soil profile is such that it holds water a little bit longer than other types of soil. And so we don't need to water quite as often. So these are things that we can look at as well. The leaves can yellow and die, again, from the inside of the plant outward and bottom up. And also premature leaf drop. So we can see all of those things associated with it. So stem girdling roots, these are when we can see these roots growing around the tree. You can see that in this picture in the middle as well. In this lower picture, you can see the roots have just kind of grown around and around this particular tree. And what happens is oftentimes if people are purchasing plants and they've been in the pot too long, then there could be the potential for those rings to go, or for those roots to grow around that pot. And then if they don't get spread out or clipped a little bit to, pruned a little bit before planting, they'll just continue to grow around and around because that's all they know. It could also be that the planting hole is too small and narrow. So there's no room for those roots to start spreading out. Soil texture differences. We used to, a long time ago, say that we should amend the soil when we're planting our trees, but we no longer do that, because what happened was we started seeing more girdling roots, because the trees would start growing in that nicely amended soil, and then they'd hit this clay wall, because a lot of our soils in Colorado truly are clay, and the roots hit that clay wall. Well, they don't want to go through that. They want to keep growing in that nice amended soil, and so they're just going to keep growing around and around and around, and that causes girdling roots and eventual tree death, because the trees just can't sustain themselves like that. So we have to be careful with all of those things. So how do we identify stem girdling roots or girdling roots on a tree? Well, oftentimes what you'll see is the lower part of the trunk, you'll see at least one part of this trunk is flattened, or you may see bulging on other parts of the tree. So in this lower left picture, you can see both this flattened side as well as this little bulging area down at the base. In this picture on the right, you can definitely see the bulging root, or the bulging lower trunk down here. You might see something like leaf scorch or dieback. You might see smaller leaves. You may also see that the whole plant is smaller itself, off-color foliage. And, how do we know that these two trees were not planted at the same time? And this one's declining because it wasn't planted correctly and it's got girdling roots. We don't know that for sure, but that's a possibility. It may just not have grown very well. So all of these things could be symptoms of girdling roots. And then also, getting a good picture, and you can say, oh yeah, girdling roots right there. So oxygen starvation, again, compacted soil, impermeable or slowly impermeable soil coverings. So for example, landscape fabrics. We often think of these as good and beneficial. However, we may want to start looking at these and saying, well, maybe not, because you put down the landscape fabric, and if the hole isn't big enough, you can have it can actually cause girdling around the lower portions of these plants, like this shrub right here. And also soil blowing in, or sand or anything blowing in there, can clog up the pores in this landscape fabric and reduce the water availability to this particular plant. So you have to be careful sometimes with these things. I know that they can be good to reduce weed problems. People don't like to go out and pull weeds. I get that. I don't either. But sometimes these can do more damage than good. So it may not be the best thing. Somebody piled all of these lawn clippings right up against the base of the tree. Number one, any type of mulch you put around a tree should be pulled away at least 6 inches away from the trunk of the tree, so that it's not right up against the trunk and can't cause trunk damage. But number two, this is such a huge pile, and it's not going to dry down immediately, but it's going to stop any water that's going to try and get down to the roots of the plant. So these are things that we need to watch out for as well. Mulch volcanoes, they can trap moisture in there, so they can waterlog the soil, or they can shed water so that no water gets in there. They can suffocate the vascular system or the phloem under the bark of the tree. And if you put this much mulch on it and you've not planted it correctly, you might get roots that actually are going to start growing towards the surface of the soil and actually try and grow in this mulch, because, hey, it's a lot easier than trying to get through the soil profile, right? So you might have that. And again, you can get rodents or other, animals in there feeding or nesting, things like that. Another thing that I want to caution against are these rubber tree rings that people put around their trees as mulch. They retain a lot of heat. They do not allow for any evaporation of water, so they can actually suffocate the tree as well. So I don't recommend those as a plant pathologist. They're just not very good for the health of the tree. So mulch volcanoes are bad. Proper mulching is good. So you need to have a little bit of space between the mulch and the base of the tree, and then kind of move it out. So it's, in that 2-foot diameter, or however you want to do it. So too much water. If you see something that looks like this, yeah, probably not a good idea. And these types of bathtub rings around trees are not advisable either. I know that they do this. A lot of people will do this for convenience, because they think, well, I can just put water in there, it'll go down into the roots and I'll be okay. But when you run into a situation like this where you're not getting a lot of drainage, that could just kill your tree. Because there's too much water in there. So you can see things like that. And especially if you've got geese or ducks floating around in there, you kind of know that that's too much water. And here's just another example of poor soil drainage and overwatering. You've got this stagnant water just sitting here, and you can see the footprints in here where the water is out here. It's not draining through the soil in this particular example either. And so they've kind of got this bathtub ring built up, and this isn't good for the plants. Not going to be good for the trees overall. Flooding can also cause problems. Even though we think of flooding as fast moving water and maybe not doing as much damage as standing water, really flooding can move a lot of sediment. And even short-term flooding can cause damage to the trees, because it's moving soil away from the root zone. So that soil profile that is supposed to be stabilizing the roots gets moved, and all of a sudden the tree's not knowing what to do. It may have too much water, not enough oxygen. And so all of these things can contribute to causing problems. So the flood in 2013 actually caused a lot of problems down Boulder Creek and into the, going into the St. Vrain in that September when, all that water came down all at once, and it moved a lot of soil and it caused a lot of damage to trees that were along some of those areas at Boulder Creek. So, again, even though it seemed to be short term and the water wasn't that high for that long, it still caused a lot of problems. Planting too deep can also be a problem with trees. So we can have too deep or too high. The picture on the right is too high, where you have this. Yes.
Do you want to take a break for a little bit and we can come back? Let everyone get up and have a stretch.
What time is it?
10:30.
Oh, yes. Yes, please. Sorry. I wasn't paying attention. Okay. Okay. Not a problem. I will stop here and we'll come back to this.
All right. Sounds good. Provided Tamla is back. Tamla, are you back? All right, we will wait until Tamla gets back, and then once she's back, we can get started again. So we might have another minute or two.
Hey, Cassie.
Hello, welcome back.
Yes, thank you. Good news. We're next on the list to get plowed out.
Oh, I wondered if something had happened. All right, cool. Well, we are ready when you are.
The little things in life, right?
Indeed.
Okay, you need to stop sharing your screen.
Right. No, I think I did.
Did you? Okay. Well, because I still see the what, cabbage seedlings?
Oh no, that's just my picture in the background.
Let me see.
Those are peas. It's St. Patrick's Day, so
oh, okay.
it'd be seasonal.
Peas, awesome. Okay, let's see. Oh, here we go. Okay. Got it. Sorry about that. Thank you all for being so patient with me today. I do appreciate it. So, okay, so we were talking about oxygen starvation caused by too deep a planting and no root flare. So a root flare is actually at the crown and we see it. And the picture on the right is an example of planting too high. This is a little too much root flare, and this is none on the left. And so we want to make sure that we have that happy medium and we have that correct root flare. Tree planting depth, again, talking about something like this, there's no root flare in this picture on the upper left, too much root flare in the picture on the right, and the picture in the middle, we have a mulch volcano. So we really can't see how that was planted, but judging by the color of the leaves and how thin the crown is, as far as like how thin the leaf material is on that, I would say that it's probably not planted correctly. So some of the tree damage symptoms that we see from oxygen starvation are things like iron chlorosis, crown dieback, early fall color, poor growth. This is actually a picture of interveinal chlorosis on maple, and this is actually caused by iron deficiency. And we call it interveinal chlorosis because we have the green veins that go down the middle, but the leaf tissue itself is turning yellow. And this can also be an example of early fall color. This looks like a calorie pear or one of the ornamental pears. And so you can see that the tree's turning yellow, but it's not quite the right time for it yet. So how do we manage this? Well, determine the cause and make corrections. That being said, if it's something like a mulch volcano, yeah, move that mulch away from the base of the plant. Sometimes corrections are not possible though, simply because if the tree is planted too deep, we may not be able to lift it up. If it was newly planted, like it has not established, like you just planted it, a week or a month ago, you may be able to pull it back up and then replant it and have pretty good success with that. But if it's an established tree, we're going to have a problem because there's no way that you can pull that up. The best thing that we can do is try and improve our soil drainage, aeration. If your tree is in a turf situation, you may be able to core aerate, do some core aeration and maybe add a little bit of compost in there so that, the compost can add some, soil tilth to it and to add a little bit of texture to the soil so it will drain a little bit better and we can get some good aeration in there. If you have black plastic, remove it. Black plastic is not recommended for landscapes. It's just not feasible. So I highly recommend getting that out of there. It's water can't get through it. Weeds can grow on top of it. It traps moisture in there if you have a whole lot of moisture, and it's just not a good thing to do that. Pull the mulch away from the base. As I said earlier, you could do that and reduce the mulch thickness. Don't make it so thick. I know it's kind of a pain, but it is our landscape. And so if we put, we always want to do too much at one time, and we want to make sure that, we don't have to go out there and add a whole lot of mulch later, but really we need to. Just put down the right amount of mulch and just add a little bit every year for what either blows away or decomposes. All right. So we have wind damage that can happen. Like on spruce trees or pine trees, we can have the branches rubbed together and it can rub the needles off. And so some people think, well, what's happening to all of my needles? What's going on with that? And we can look at, the winds, whatever we're supposed to do, things like that. Any kind of tattering might be available. Could happen to the plants as well. Out on, say, an more open area like the Eastern Plains or something like that, where when the wind blows, you get a lot of soil movement or sand movement, and it can also be almost be like sand blasting the trees and causing problems. So again, it's one of those things, something that could look like that. Okay, nutrients. Let's talk about nutrients. So we have our macronutrients. These are the nitrogen, phosphorus, and potassium. They're needed by plants in relatively large quantities. Now, our soils generally have enough phosphorus and potassium, although we can add a little bit to the soils. But plants, especially in our garden, the most common nutrient that we need, very mobile nutrient, and plants use a lot of it, and it moves through the plant fairly quickly. And so we have to keep, look at our garden and keep checking on it to make sure that we are offering enough nitrogen for our plants. The micronutrients, these are things like calcium, sulfur, iron, magnesium, manganese, boron, and zinc. These our soils contain sufficient micronutrients most of the time. There are some exceptions. although, they contain it may not be available to our plants. And so we're going to talk a little bit about that, especially in terms of iron. So nutritional deficiencies, again, like I said, they're highly mobile in plants for nitrogen. Plants will grow poorly. Nitrogen deficiency starts with lower leaves on the bottom portion of the plant. And so the lower leaves will start to turn yellow or light brown, and then it looks like it kind of moves up the plant. Stems are short and tender. The thing with nitrogen, though, especially in our vegetables, is that we don't want to overdose on nitrogen, because if we're trying to get fruit, say in our tomatoes, that overdose of nitrogen can actually move through the plant and bypass the fruit. And so the fruit's not going to grow very well. And so we just don't get any fruit set. So it's one of those things where we have to monitor that and make sure that we are doing the correct thing for our plants. Calcium, nitrogen, or calcium deficiency often exhibits itself in our vegetable garden as blossom end rot. This is where the plants are not getting enough calcium. Calcium is actually a component of cell wall construction in our tomato fruit. And so if the calcium bypasses the fruit, the cells are not, don't have enough structure in them to support the growth of the tomato. And so they just start decaying. And it looks like a disease on the bottom of the tomatoes, but it's actually just a calcium deficiency. Now, the leaves on the plants can also show some hooked tips or curled margins as well. The terminal buds may die, and the plants have a really poor root system. So again, this is one of those things where we caution with blossom end rot to make sure that there's calcium in the soil. Now, calcium in the soil is important because calcium has to come up through the root system of the plant. Calcium sprays do not actually work, other than what they're doing is it runs off into the soil, the calcium goes in the soil, and then the roots will take it up. But calcium sprays don't actually work like that, because the plant has to take in the calcium through the root system. So again, if we monitor, good consistent watering is important for that, to make sure that that calcium is coming up through the root system of the plants. And tomatoes are very susceptible to this. Peppers can be as well. And even cucumbers, some of our cucurbits are susceptible to blossom end rot. So we'll have to be careful with that. We do want to avoid too much nitrogen fertilizer, because that nitrogen, as it's moving up in the plant, because it's mobile, will pull that calcium with it. And so it pulls that calcium and it bypasses the fruit. So when we talk about monitoring for early blight in tomatoes, and we say that, you need to watch your nitrogen in tomatoes, because early blight is usually exacerbated by nitrogen deficiencies. You don't want to add too much nitrogen, because that's going to bypass the fruit, and it's also going to pull the calcium past the fruit. But you still need that nitrogen in order to reduce early blight in tomatoes. So again, it's a balance. We have to watch out, and we have to make sure that things are going well, and that we're not that we're monitoring our fertilizers and not getting too much or too little. Here's an example. Again, this is a melon with blossom end rot. And one of again, like it said, calcium has to come up through the roots. It's not, it doesn't come in through the stomates or any of the lenticels on the stems. And if we keep our soil moisture even, that allows for more consistent uptake of these nutrients to our plants. And so it avoids some of these problems as well. Iron chlorosis. This is where the plants are not taking up enough iron. They're just not getting iron. Now, the main veins will remain green, but the rest of the leaf tissue will turn the yellowish color. And so we want to make sure that we are monitoring this. Sometimes you can get browning of the leaves because, of course, when the leaves turn yellow, they're not photosynthesizing properly. And so it's, if they're not photosynthesizing properly, there's no nutrients in there, there's no energy. And so that tissue will just start to decline and turn brown and it will die. And sometimes leaves will be shed. Now, the thing about iron chlorosis is that it affects the younger leaves first because the iron is not getting up into those new leaves. And so they are showing that symptom from iron chlorosis. Some of the older leaves may look normal, but the newer leaves will look chlorotic. And susceptible plant species can aggravate iron chlorosis, like some of our maples, some of our oaks, different things like that. Cool soils, conditions that restrict air movement and oxygen movement in the soils, poorly drained soils. So of course, oxygen starvation, poor drainage, too much water, compaction of the soils or use of plastic mulching, even, taking topsoil off and leaving just that lime enriched soil there. Planting close to house foundations can do that simply because you could get some of that lime leaching out from the cement foundations on houses. That could aggravate iron chlorosis as well. Some of the soil amendments available for iron chlorosis can be acidifying nitrogen fertilizers like ammonium sulfate, even some elemental sulfur. It might work, it might help the plant, but to put in a whole lot of sulfur to lower pH by 1 unit, the pH of the soil, that's not going to happen. So again, we have to make sure that we are, watching this and keeping it. Iron sulfate might help, or a combination of sulfur and iron sulfate. But this is what I was talking about, where the leaves themselves will start to turn brown. The veins are green, but the leaf tissue started to turn yellow, and as such, it's not photosynthesizing properly. And so consequently, we're going to get those brown areas in there. So our nutrient deficiency symptoms, this is just a very generic chart, very minimal, but it can kind of help you look at your plants and determine, well, maybe I have a nutrient deficiency. Are the symptoms on the lower leaves or the upper leaves? So this is kind of a key to walk you through that. So upper leaves and lower leaves, and then looking at the color pattern. Are the internodes short? What color are the leaves? Things like that. And so we can also end up at like nitrogen deficiency, potassium, phosphorus, things like that. So this one over here is sulfur deficiency, zinc, iron. And of course, it's not all of the nutrients on this chart, which is why I say it is, kind of a good starting point, but it's not necessarily the be all end all. And one of the things also to look at is soil pH. So what you can do for landscapes is recommend a soil pH test, a soil test that will tell you how much organic matter, how much nutrients you have in your soil, what kinds of nutrients, what you might need to add to your soil, depending on what you're going to plant, and then the pH of the soil. So for example, this particular chart actually shows you, so strongly acid 4.0 goes to 10.0. And if we look at iron specifically, iron is available to our plants until about 6.5, 6.3. It starts declining in availability to the plant. By the time we get up to 7.5, 8.0, 8.5, it is extremely restricted in our plants. Basically, what happens is the iron precipitates out of the soil at that high pH. And when it precipitates out, it is no longer a salt, liquid, or it can be taken up by plants, and the plants can't take up a precipitate. So again, it can't take up the iron, even though our soils have a lot of iron. The pH actually binds the pH of the soil. It binds that iron to the soil particles and precipitates out, and so it's not available anymore. So how do you manage it? Again, with those acidifying fertilizers, ammonium sulfate, things like that, you can do that. Now, for high value plants, high value trees, what about trunk injections of iron? And that is a possibility. You would have to talk to a certified arborist about those who is familiar with injections in the trees in order to see if it's worth it, you know, if you want to do that. It can work for a couple of years and then you might have to do it again, but it may actually help a lot. The problem with adding iron supplements to the soil is that it will, that iron will get bound up by the pH of the soil again, so it's not going to be readily available. So, you know, sometimes we used to say like chelated iron supplements you could use, but again, they're only minimally helpful. So yeah, you have to kind of look at everything as far as what's happening. Chemical injury. Okay, so I'm just gonna, I have a few cautions about diagnosing chemical injury. Now, a chemical injury could be an insecticide, it could be a fungicide, could be a herbicide, whatever kind of chemical it is. It could be a homemade concoction that somebody made up. Unless the client is willing to have the plant tested for any chemical injury, there can never be a positive diagnosis. When talking with our clients, the terminology is always damage appears consistent with, but we don't know for sure unless you want the plant tested. And testing for chemicals is expensive. There are testing labs out there that can do it, sure, but it costs money. The last time this has been about 4 or 5 years ago, the last time I got a quote from one of the labs that I used to use, they were charging $175 for a phenoxy screen, and that was like 2,4-D, dicamba, things like that. It was about $350 to test for glyphosate. Aminopyrrolid test was somewhere in the neighborhood of $300 to $400. So it is not cheap, and that's only for one plant sample. So again, unless the client's willing to pay for that, we never give a positive diagnosis. All we can say is that the damage appears consistent with. So for example, in this particular picture, say, well, it looks like it may be, just because these needles are overgrowing. They're very thin. They don't look like the rest of an arthropod called areophyid mites that can do damage that looks like this. So we cannot ever jump to it's a chemical injury. We have to do our due diligence and diagnose the plants and make sure that we're not finding anything else before we say, well, it looks like it's consistent with something or another. This particular one, it could be like that Phyton 27 damage picture that I showed you earlier. You could have this loss of leaf color. You could get wilting. It just depends on what's happening. Oftentimes with herbicide injury, there's twisting and curling, distortion of leaves, and then you see these new leaves that are coming out are all twisted and curling. So this looks consistent with phenoxy herbicide damage, and phenoxy herbicides are growth regulators. And the same thing over here in this picture on the left-hand side looks consistent with a phenoxy herbicide. Chemical injury, this particular picture on the left-hand side looks consistent with dicamba, which is a very common active ingredient in a lot of our weed and feed products that are sold. And so, that leaches readily through the soil. And so it can, show up in the plant as symptoms that look like you have this exaggerated growth of this particular leaf that could be consistent with a phenoxy herbicide as well. And then we have other things that look like you get these fan-shaped type leaves. You could have white growth tips. You could have purpling color on spruces. And that's really an interesting one to diagnose, and be caused by phosphorus deficiency, cold temperatures, drought, some types of herbicides. So you can't ever jump to the, oh, that's herbicide. So that's why we say you can't ever jump to that conclusion. Other times you might see things that look like this, where these are the leaves that are coming out. This was actually dicamba again that was applied under a tree's drip line, and so it was taken up by the plant and caused damage to the newly forming leaves. Sometimes we will see upward cupping of the leaves. Sometimes we'll see twisting or epinasty of the petioles and stems on the leaves. Like this lilac right here, it's very not very distinctive for this epinasty associated with this, but again, it can be. So, you know, we're looking at things that look like this. This is epinasty on tomato. You get this curling of the stems. You get the leaves that kind of curl over and things like this. This was actually a sample that I worked on. I never tested it for chemicals, but I was pretty sure that it was consistent with some sort of phenoxy herbicide. But again, you know, all I can say is, well, this is what it looks consistent with, and go from there. Glyphosate damage on plants. Glyphosate actually will, if it's sprayed on plants that are dormant, it can affect the growing points the next season. And so consequently, you get this kind of witch's brooming look associated with it. So that could be a potential one for that. You can also see leaves, so you know, where the leaves are just kind of they don't look normal. They're the edges look kind of feathery, but they're not exactly like they're supposed to be. We can also have chemical injury. This is a phytotoxicity associated with English ivy, I believe. Somebody sprayed the concoction of baking soda and water on the plant to kill something, and it's toxic to this particular plant. So again, this was a homemade concoction, not recommended. You know, we use scientific recommendations, research-based, and this was not a recommended product, and it got sprayed on this plant, and this is the chemical damage associated from that. So again, it's not always a pesticide, but it can be a chemical. Now, some things can look like herbicide injury, like this picture on the left. This is an apple tree. This looks like somebody may have sprayed it with a herbicide, but this is, if you were to pull this leaf off, look on the underside of the leaf, it looks like little blisters. And if you open, you know, just peel it open, you would see those little, I mean, and we're talking microscopic areophyid mites. You would need a good hand lens to see them, but that is what happens there. These two pictures are actually honey locust plant bug. It looks like a herbicide injury, but this is honey locust plant bug damage to honey locust trees. And I see this all the time in the, you know, spring and summer, what happens with that. And, you know, then we have hackberries that get hackberry nipple gall. That looks like it might be a disease gall, but it's actually an insect that is causing that damage. And then, you know, we can see things that look like, well, maybe they have a virus disease, or maybe they, you know, there's some other abiotic stress, nutritional deficiencies, not necessarily iron chlorosis, can look like, you know, herbicide injury, but it's really not. So this is why we have to do our due diligence, ask our questions, and figure out what's going on with the entire plant before we can make a definite diagnosis. This particular one in the lower left, this is magnesium deficiency in apple. So again, not a herbicide problem, but it could look like it, like these tips are dying back, and you know, somebody may have treated it with something that they shouldn't have. So in this picture, I put this up here to kind of practice. And, you know, if you guys are looking at pictures and you're looking at a landscape and you're wanting to look at this, you might say, okay, well, this looks like a pretty nice garden, right? And so you look at this, and a person who sent me this picture had, you know, what was wrong with their plant. And I was like, well, I suspect it's a herbicide. I tested it for virus issues and it was negative. So, you know, the common landscape viruses that we see. And he sent me this picture and I was looking at it and I looked at this edge garden right here and how this is all brown. And so I emailed him back and I asked him what this brown edge of the turf was, right? You know, along, I mean, it's a very straight line. And he said, well, he had some, the turf was growing into the landscape garden here, this perennial area. And he had some weed killer in the garage. And so he sprayed it on the turf. Well, okay. That just tells me it probably was a herbicide injury to the specific plant that he was asking about. But again, you know, people think, you know, sometimes that what they do isn't going to affect other things. And so we have to be very careful when we use chemicals. It's, you know, things like this can happen. And so we have to be very careful. So again, just to recap, for abiotic disorders, we have the plant, there's some sort of interaction between the plant and the environment, and that can cause a lot of problems. And so we want to make sure that we are, you know, monitoring that and not, you know, and taking action if we can. I'm learning as the older I get that there are some things I just don't have control over and I just need to let them go. And sometimes the environment is one of those things. I live in an area where the abiotic issues can be extreme and I can't control the weather. So nothing I can do here. So anyway, do y'all have any questions before we start moving into the biotic issues of plant problems, of plant pathogens?
The only one I had wasn't so much in question as a request for the nutrient deficiency chart in a PDF form.
Okay, I can do that. I'll send it to you, Cassie, and you can disseminate it out to the other coordinators.
Perfect. Thanks so much.
Uh-huh. Do they want the pH chart too?
I did send the link, but we might as well. Yeah.
Okay, I can do it. All right, sounds good. Okay, so we have about 15 minutes until we go to lunch, right? So I will get started with the biotics and then we will continue after lunch. So that'll be good.
Sounds like a plan.
So we talked about the abiotic issues of, good. We talked about the abiotic issues of plant problems. Now let's talk about some biotic issues. And biotic, as you remember, are the living causes of these problems. And we're talking about microorganisms because you can't see them without a microscope. And some of them you can't even see with a microscope. You're just not able to. So we'll talk about some of these as we move forward. And the two lower pictures here are both abiotic issues. Somebody put these plants in here and then it looks like they just did not get watered because they planted over here and these plants look really good. Picture on the left is blossom end rot, and we already talked about that. That is a calcium deficiency. So this picture in the upper right, African violet. Now, I don't know what's going on with this, but this looks more like a disease issue to me. So we will start investigating. So at that point, we start investigating on it. So the biotic causes of problems. These are the living organisms that cause plant problems. So we talk about fungal diseases. We're going to talk about bacterial diseases, virus, phytoplasma, nematodes. I talk about parasitic plants only in the sense that we live in Colorado, and some of you who may be listening may live in the mountains like I do, or in the foothills, and you may run into mistletoe, and that's a parasitic plant. And so I talk about that only in that sense. In landscapes along the Front Range or out on the Eastern Plains, chances are you won't see those simply because it's just not the right environment for them. And the right host plants are not there as well. Other biotic causes of diseases, and that's only because they are living organisms, are animals. I'm not going to talk about those, but things like rabbits, deer, in my case, deer, moose, bears, whatever happens to walk through my property is, those are animals that I all have to deal with. Voles are a huge nuisance up here. Insects, that's a different presentation. Whitney gave that one. But they're living organisms as well, and they can cause plant problems. So we can talk about all these different types of living organisms associated with it. So fungi and bacteria, these are probably going to be our two main ones. These are spread by wind or water, soil movement, animals, including us. We can move them around. It can move on equipment, plant material. Insects can move these as well. For example, Dutch elm disease is a fungus in American elms. Ophiostoma nova ulmi is the pathogen. But the banded elm bark beetle is now the primary vector or mover, spreader of that Dutch elm disease. So we have to, monitor that particular problem in order to manage Dutch elm disease. We can, wind can blow spores, water, even splashing water if we're doing overhead irrigation in our gardens can be a problem. Driving rain, snow, windstorms, soil movement can cause problems. So again, all of these things can be spread by these particular issues. So we want to make sure that we are monitoring that. Fungi and bacteria both get into plants through natural openings on the tree. Now, there are some fungi that are what we call active invaders, meaning that they can actually penetrate the plant itself in order to start the infection process. And powdery mildew is one of those. Bacteria are what we call passive invaders. They actually need to have a wound or in that way too, but oftentimes fungi are going to be a little bit more active about penetrating into a plant. So bacteria can get in through stomates, lenticels, insect wounds, different sort, you got birds, woodpeckers damage, things like that, some of these pathogens can get into as well. And so what these pathogens do is they produce enzymes that break down the cuticle, and then they can start getting into the plant, into the vascular system, and start moving within the plant and start growing basically inside the plant. And fungi and bacteria cause a lot of the same symptoms, which is why it's really difficult to diagnose, because you have to really do due diligence, like I said, to make sure that you are diagnosing these properly. So we have leaf spots and curlings, we have galls, soft rots, wilts, cankers, stem and root rots are all caused by both fungi and bacteria. And so consequently, we see leaf spots that look like this. This looks like black spot of rose. Well, that's a fungus. This leaf spot on the geranium could be Pseudomonas, could be botrytis, could be Xanthomonas bacteria, Pseudomonas bacteria, botrytis fungi. We just don't know until we start doing the diagnostic process, looking for spores, things like that. And so, and it makes a big difference, because if you're going to treat these plants, say this is a greenhouse situation, and you want to treat these, if it's a fungus and you treat it with a bactericide, not going to do a thing, and vice versa. So you have to make sure you diagnose the right problem. Root rots, again, stem and root rots, there's several different fungi and bacteria that can cause these problems. So diagnostics is really important, and making sure that we know the pathogen before we start treating. Fungal characteristics, there's a variety of sizes of fungi, and by sizes I mean different sizes of spores and different colors of spores. This particular spore in this picture that this arrow is pointing to is actually an Alternaria spore. Now, Alternaria, you might say, well, what the heck is that? Alternaria is the pathogen that causes early blight in tomatoes and peppers. However, early blight is Alternaria is also an opportunistic pathogen. So consequently, it's kind of hanging out there. Alternaria is also one of those spores that when they do spore counts on the news that you see, pollen counts, spore counts, things like that. This is one of the fungi that they count for spores because these fungi are just blowing around. But these can aggravate hay fever sufferers. So if you have hay fever, you might want to watch, the spore counts and see what the spore counts are, because that could aggravate it. And a lot of these, again, like I say, they're just hanging out on a plant. They're what we call saprophytes. They're not doing any damage. They're just hanging out, having a good time, not doing anything until the plant starts getting stressed. And then these Alternaria will become a pathogen. So then they start doing some damage to the plants. So then we have to watch it. Most fungi reproduce by spores. However, there are certain fungi that do not produce spores, that only produce mycelium. So it just depends on which one we're looking at and what we identify and how that goes. So usually with fungal pathogens, and you'll notice I'm saying usually again, we have these distinct margins that between the area where the fungus is growing out into the leaf surface and where the fungus has already colonized and has done damage, and we have this necrotic lesion. So, and usually we'll have some sort of halo around a fungal lesion like this, but bacteria can also exhibit this symptom sometimes as well. So this is why it's really tough to be a plant pathologist and to diagnose these things. It really can be a challenge to figure out, okay, what am I dealing with here? So in this particular case, we have a fungal pathogen that has this margin edge around it. And so we are looking at this specifically. So there are some exceptions, of course, because things like apple scab does not have a distinct margin, nor does black spot of rose. They have what we call feathery margins, meaning that there's not really, this has a halo around it, but there's no definite margin like that was in the previous picture. So we have this distinct margin from the previous picture. And in this picture, no distinct margin, we have the feathery edges. So again, those feathery margins. So we see those. Fungal ID tips. Well, for fungal ID, and this is what I tell my students, is fungi are fuzzy and friendly. Bacteria are slimy and smelly. Not always true, but for the most part, that's a good generalization. Strawberries, if you see that gray mold that's growing on strawberries, that is a pathogen called botrytis, and it's fuzzy, right? Oftentimes you'll see distinct borders associated with fungi, but again, like that black rose or black spot of rose and the apple scab, you won't see that. And oftentimes the damage is random. So if you remember back to when I was talking about leaf scorch, and we had the margin edges of the leaves were all fairly evenly browning from the tips back, this is not that leaf scorch. This is Marsanina of aspen, Marsanina leaf blight. So again, these spots are random. They can keep growing and they'll grow together. And so you see big blotches like this. But again, it's one of those things to look at. I'm going to stop here. And then when we come back from lunch, it's 11:27, and we come back at 12:15, right? And
technically
then
we go until
I'll start up with the individual pathogen.
Technically we can go 11:45 to 12:15.
Okay, I thought it was 11:30.
If it's up to you.
Oh, I thought it was a 45-minute lunch.
It's on Katie's schedule, it's half an hour. But we are a little bit ahead, so if you want to. Going. Okay.
Nope, we'll keep forging ahead, and we will definitely probably end up early today then. Okay, good. Okay, powdery mildew. When we were talking about symptoms and signs, I was talking about powdery mildew as both a symptom and a sign, because when you're looking at the plant, you see the symptom of the whitish powder on the leaves. When you start looking closely, it's a symptom because that's the actual mycelium that is growing on the plant itself. So it looks like somebody sprinkled flour or talcum powder on the plants. They are host specific, like I mentioned. The powdery mildew on your rose is not the same one that's going to go to your lilac and vice versa. In Colorado, it's very common. Oftentimes we think of mildews as having to have needing a very moist area, but that's not true. We in Colorado, we are pretty arid. Powdery mildew needs a little bit of humidity, but it doesn't need free moisture. And so consequently, when our plants are really close together and things like that, it builds up that humidity in the plant canopy. And that's what can encourage the powdery mildew to grow. It's not necessarily that we have a lot of free moisture. It's not necessarily that we're overwatering or anything like that. It's that the plant canopy itself can produce that humidity. So that's what we have to watch out for. So it can produce a little bit of discoloration. There might be some reddening associated with powdery mildew, and that depends on the plant host. There's also some distortion, as in that lower picture on the apple. And it can also be premature leaf drop. It doesn't necessarily kill the plants, simply because powdery mildew is what we call an obligate parasite. And this means that it needs a live host in order for the pathogen to survive. So when the plant is going dormant or senescing for the fall, this is when the powdery mildew senses that the plant is no longer producing all the nutrients, photosynthesizing everything that it needs. So that's when it starts producing those cleistothecia, the overwintering structures here in Colorado. And so that's, I hate to anthropomorphize plant pathogens, but they really do sense that the plant is going dormant. So they're producing those overwintering structures so it can survive, and then it can reinfect the plant in the spring and continue to grow. So it never truly dies out. It's always there, and it has to have that live host tissue in order to produce the mycelium. So for powdery mildew management, again, we look at, you can use things like neem or horticultural oil. Fungicides, there are some available. There are sulfurs that you can use. With powdery mildew management, though, you need to make sure that if you're using sulfur, read the label because it is very temperature sensitive. So if the temperatures are going to get really hot during the day, don't use the sulfur. You could end up burning the plant, and not like burning, setting it on fire, but the phytotoxicity. You could end up with the leaf damage associated with that. You can spray the plant with water to maybe wash the powdery mildew spores off of it, but that doesn't always get rid of it because powdery mildew actually grows intracellularly. So it's actually growing inside the plant, and it's producing the spores and the spore structure that holds those spores from the inside of the plant to the outside of the plant. So even if you wash it off, you've just slowed it down for a little while. It's still going to produce those spores. It'll come back. Potassium bicarbonate is another option that you can use. Do not use sodium bicarbonate, as that picture that I showed you in the abiotics. You can cause phytotoxicity damage. And as well, sodium is not necessarily needed in our soils. We don't need any additional sodium. So what are some cultural things that you could do? And here's that baking soda burn picture again. Cultural things that you can do are, again, proper pruning of the plants. Make sure that there's some good air circulation around them. Site your plants, put them in the right place so that you don't have dead air pockets, so you do get some air circulation. Avoid over fertilization because the pathogen just loves nutrient rich plant material. So because it keeps feeding on all of those nutrients, avoid overwatering. Keep the water out of the plant. Water early in the day, good sanitation. removing any of the infected material and raking up fallen leaves. And that's because you don't want that leaf tissue that has those Cleistothecia to overwinter, because then they're there to infect the plant in the spring. So again, it's one of those things where we need to look at that. You know, just good proper doing a lot of good remediation for these pathogens. Marsanina leaf spot. This is on aspen and some other poplars. Basically, it's a very common plant pathogen. It's mostly aesthetic. Again, this one's not going to outright kill the plant, but it may lead to some chronic stress, so the plant is not as healthy as it should be. And these spots will be various sizes because they can grow together or coalesce. And so you may see larger spots versus smaller spots, and so on and so forth. Infection occurs at bud break, and then you'll see these show up about midsummer. So along about July-ish, usually you'll see these symptoms start to show up on the leaves. But the infection started as those leaves were coming out. And so treatment for that is to make sure that if you want to treat for that, that you use a fungicide at bud break. You can use chlorthalonil as the active ingredient, and it's a protectant. And so it will protect those leaves from being infected by the Marsanina pathogen. Now, you may have to use a couple of treatments if you want to use a fungicide. Otherwise, again, good sanitation at the end of the season, raking up those leaves so that those spores are not available to infect the plant in the springtime. So you might be asking yourself, can I compost those leaves or any other diseased plant material for that matter? Well, that's a good question. The compost has to be hot compost, at least 140 degrees, consistent throughout the pile, to kill most of the disease organisms. However, most compost piles are not going to be like that, and disease organisms are everywhere. So what I recommend is that not composting your diseased plant material, simply because, especially if you're getting rid of materials that are infected with a virus, and that virus is moved about by insects, then if that plant does not completely break down, the insects can still be feeding on those plants, pick up the virus and take it, and most of them fly, so they could take it to another plant and infect it. So I know it's not what we would consider to be sustainable, but by the same token, you don't want to keep bringing disease problems back into your landscape. And even using chipped mulch from a diseased plant. Dr. Jacoby, before he retired, he and a research associate that worked for him did a study on Thyronectria canker in using it as a mulch. Honey locust trees that had Thyronectria, using that as a mulch around trees. And what they did was they chipped up diseased trees, and then they used that mulch and spread it around. And they went back like 2 or 3 years in a row to pull out wood chips that were infected with Thyronectria. And they were still able to culture that to get the Thyronectria to grow in an artificial culture, which means that that pathogen was still sustainable. I mean, it was still alive in that mulch. And so it could have potentially infected the tree again if that tree that they put the mulch around was a honey locust. So again, you know, these are things to think about when we're doing that. Cytospora canker is our #1 canker causer, and it goes to cottonwood, aspen, willow, spruce, peach trees, apple trees. It goes to all sorts of trees, ash trees. You know, we're looking at this as a huge problem. It also is a weak pathogen, meaning that it is there, not usually causing a lot of damage until the plants get stressed. So especially for our trees, putting them in the right spot, making sure that they get good cultural care can go a long way to reducing the infection in the plants. So again, looking at that, and you'll see the cankers associated with this. You know, you can usually tell the darkening color on aspen is really distinctive. I can drive down the mountain roads and the bark gets bright orange. So it's one of those things where it's easily identifiable. And a canker is that dead tissue, right? Usually it's somewhere on the main stem, not generally going to be on a branch. So you really can't do anything about that. So fungal cankers also contain the fruiting structures of Cytospora. And when these fruiting structures start producing the spores, they can get moved around by birds or wind or splashing water. All of those things can do it. And so it's really, you know, some of these should just be taken out just because you don't want them to infect everything else. Here's a picture of the fruiting bodies, fruiting structures embedded in the bark. You can see, I showed this picture earlier. They look like little pimples or little mini volcanoes growing out of the bark itself. And what happens with these cankers is they actually disrupt and kill the tissue beneath it. So there's no water conduct system in there. So it just really can't get up to the tree, and it causes a lot of problems. Excuse me. These are the orange spore tendrils that are produced by those fruiting structures that we saw in the previous picture. And there are millions of them in here. And so again, these can get, you know, birds can be on the trees. They can pick them up with their feet, their beaks, whatever. They can get blown about. They can get splashed about. Now, not every single one of these spores is going to land on a plant and cause a disease. However, it can be a problem when we start looking at these things. Colorado spruce with Cytospora canker. It looks different. Cytospora looks different on spruce than it does on the deciduous trees. On Cytospora, it often looks like a gummy canker that looks like this. And it could be on branches, it could be on the trunk. It just depends on where it comes from, but it has the same spore types. So if you were looking under a microscope, you'd be able to see them. But again, the symptoms are a little bit different in deciduous trees versus spruce trees. So how do we manage Cytospora canker? We try to grow healthy trees, prune out the affected tissue, and that raises a question as to, well, can I prune that canker out of the trunk? I highly recommend not to do that. If you were to prune that canker out, basically what you've done is you've just opened up a whole new wound on that trunk, and other pathogens could land in there, more Cytospora, whatever. And that tree's just going to suffer. It's not going to be a very healthy tree. It's not going to be very good. There are no effective fungicides for Cytospora. I know that Dr. Jane Stewart is doing a lot of work with Cytospora on peach trees, and she's looking at different fungicides, but I don't believe she's found anything yet that will work, that will help with that. So again, just good cultural care, making sure that we water and do everything correctly. All right, do y'all have any questions about fungal pathogens?
I think we've been able to answer most of them.
Awesome. Woohoo. Yay. Good, good. Yeah, I don't see any open questions in there, so good deal. Excellent. Right. Well, let's stop here a couple minutes early. It's 11:43. We'll come back at 12:15 and we will pick up, and at the rate I'm going, we might be finished by 2:30. So
sounds perfect.
Y'all get an extra half hour of your day back. All right. I will stop. Do I need to stop sharing, or do you?
I'll start.
Like put your slide over mine. A good plan. There we are. Okay. All right. So we ended up with questions on fungal pathogens. Now we are going to move into bacterial problems and talk a little bit about bacteria. So one of the things is bacteria are single cell, they're microscopic. Most of them have what are called flagella. They're like these little propellers or tails off of them that help them move in little films of water. And that's how they actually kind of move into plants through stomates and wounds and things like that, is because they can move very briefly and very, not like long distance swimmers, but they can move into areas like stomates and lenticels with a film of water. So they reproduce by dividing in two. So 1 becomes 2, 2 becomes 4, etc. So they have exponential growth. They're very difficult to identify just by looking at the cells under the microscope, because technically they're not spores. So how we identify them is to look at the symptoms, look at the host, and then we also look at what bacteria can affect those particular host plants. And then if we grow out some of the symptomatic plant tissue on artificial media, we can often determine the bacteria by the color of it. Some grow yellow. Most of the xanthomonid bacteria are yellowish in color on culture. Pseudomonas are kind of a creamy color, but some of them also fluoresce. So if you use a special media that allows the pathogen to fluoresce and you put it under a UV light as it grows, you can see if it's a fluorescent pseudomonad bacteria. So, all of these things, and I'm not saying that you're going to be able to go out and do that, but you should have diagnostic resources available to you that you could have that done if you needed to. But for the most parts, again, like I say, bacteria, I generally ID as slimy and smelly. And that's because they often have this water-soaked appearance, like when you leave lettuce in the crisper drawer for too long and it gets too cold in there and the lettuce freezes and then melts or thaws out, then you see this, it's really, water-soaked translucent type of plant material. And so again, lots of things can happen like that. They are often smelly, although some people will say that they smell wonderful, that they like the smell of it. I've never been able to tell that. To me, they always just smell. And the leaf spots on bacteria are often angular. So in this picture in the lower left, you can see they're not perfectly round or sort of round like we think of fungal pathogens. These are more angular, and they're usually between the veins, although they can sometimes go across the veins, but they're usually between the vein margins. And so you would be able to, maybe say, oh, that looks like it might be a bacteria. So then start the diagnostic process and see if you could figure that out. So some bacterial examples. This is fire blight. This particular pathogen is caused by Erwinia amylabra is the bacteria. And it often has what we call strikes, so associated with it. And this is a severe dieback, or it looks like lightning struck the plant tissue. And so consequently, it strikes. Now, when you're looking at these, when you're looking at these, also, you can see that the leaves are hanging on the branches, and that's very characteristic of fire blight. The tips will die back, but the leaves will hang on. This picture, this picture in the lower left here is one that I took. I drove down the street for 10 years, for almost every day going to campus. And like 2.5 years ago, this tree started leafing out in the spring. The amount of fire blight that this thing had, it was crazy. And this picture in the lower right on this slide is actually the full picture of the tree. They had this tree pruned, massively pruned, in the fall, the previous fall. And So whoever did their pruning probably did not practice good. And then this lasted for about a year, and then they cut it down. They just took it out because it was so bad. There was no way that they were going to be able to save that plant. So the characteristic shepherd's crooking, looks like this. You get the blossom blight, fruit lack of production, or the fruit just shrivels up. And the bacteria actually kills the tissue and it reduces food production. So the aesthetics look bad. Of course, nobody wants to look at a dying tree because fire blight can, excuse me, produce cankers that you can see right here. You can see the canker margin where this arrow is pointing, and then the canker margin down here. And then this is the blighted twig. Now, the thing to remember about fire blight is just because you see the margin right here, that does not mean that's where the bacteria has stopped growing. The bacteria could be working in here. It just hasn't killed the tissue yet. So the bacteria could be all the way up into here, all the way up into the plant, and then it could be all the way down here as well. So when we talk about pruning for fire blight, and we talk about how we do this and what we do, you have to be very careful and make sure that you go beyond the canker margins in order to try and get as much of the pathogen out of some of the ooze that's coming out with the bacteria. Now, I have to also say that Cytospora canker on fruit trees will do this type of oozing as well. So it's not necessarily only fire blight that does this. So we have to be careful when we start looking at symptoms. So the fire blight hosts have to be in the rosaceous family. It can occur on ornamentals, palm fruit bearing plants. So we can do ornamental pears, apples. It does not form on prunus, cherry or plum. Although there was one report, obscure, these many years ago, of fire blight on peach. It has not been reported on peach since. So it was very weird, and it's not considered to be a host of peach trees either. So moisture has to be present. Temperature 60 to 65 degrees, and blossoms in some case for fire blight to get into plants. And that's because we have bees that pollinate the apples, and the bees can actually pick up the fire blight from the nectaries of the blossoms and move them from blossom to blossom, move the bacteria. It's not intentional, but it is what it is. They like the sweet nectar, and so the bacteria could be in there, and then they can move it around. Fire blight can also spread because it's an existing or an old infection, and it can also spread through pruning tools. And we'll talk about how to disinfest that in a moment. But the bacteria, of course, spreads. You get the blossom blight phase. It can spread to young twigs by the bees. It can migrate down the branch, so you get these cankers, and then these cankers can overwinter, and then in the spring they start growing again, and that ooze could come out of it that insects like, and they could move that to the flowers. And so it's a huge cycle, a disease cycle of what can happen. So fire blight management, of course, we have susceptible plants, the causal organism, and the right environment. Plant resistant varieties, that's probably the biggest thing that you can do. One of the things about fire blight is that if you're doing apples or pears, things like that, you can get apples that have been grafted onto fire blight resistant rootstock. And almost every apple you purchase has been grafted. And so you should be able to find out whether it is fire blight resistant or not. I do have to caution by saying that just because it says it's fire blight resistance does not mean that it is immune, and it will never get fire blight. It can still get fire blight. It just will not be as bad as if it's a susceptible variety. So, make sure that you're planting resistant varieties and doing that. You can also use treatments like tribasic copper sulfate. You could use fire blight spray. These are the one on the left is a copper product. The one on the right is a antibiotic, basically, for treating trees to prevent the movement of fire blight into the plant. The problem with these is that they're very specific timing. So you have to make sure that if you're going to use these, you use them correctly and read the labels. So, you know, once you see that your tree has fire blight, it's way too late to apply any sort of preventive treatment, or there is no cure for it. So you would not be able to apply a curative either. So proper spacing, thinning, keep the water out of the canopy, etc., is always good. Pruning fire blight damaged trees. How do we do that? Well, if it is the dormant season, I highly recommend going at least 12 inches beyond the visible infection. So if you see a margin edge, go 12 inches back and then make your cut. If the tree is not dormant, I recommend at least 18 inches past that visible infection, because the bacteria could have moved in that tree and you just don't see it. Now, between each pruning cut, you should disinfect your pruning tools. So how do you do that, right? Well, you can use like a 10% bleach solution, like 1 parts of water to 9 parts of bleach, and dip your clippers in there, your pruners in there. Make sure that you wipe that off before you prune, though. Make sure you wipe the bleach solution off, because that could also damage your plant. So you have to be careful. You could also use disinfectant sprays like Lysol disinfecting spray. You could use kills 99.9% of germs. And that can reduce it. But again, after you spray it, let it sit for a little bit and then wipe it off before you prune the tree. And there's also rubbing alcohol, 70%. You could use that, swirl it around, and again, wipe it off before you make your pruning cut. But these are, you know, a couple of different ways that you could do that. I don't know about using disinfecting cloths, you know, like the Clorox wipes or the Lysol wipes or whatever. I don't know about using those. I would imagine that if they have the same germ killing power, then you can use those as well before you prune. But again, just make sure that you disinfect your pruning tools, and that's going to help reduce the potential spread of infection. Because if you don't disinfect and you just keep pruning, you could potentially be moving bacteria to places that didn't have it before, and now all of a sudden your tree is like way infected with fire blight. So just making sure that you do that is really important. Okay, let's talk about viruses. Virus characteristics. Viruses are submicroscopic particles. You cannot see these with a regular microscope. You really have to have an electron microscope. Virus characteristics, they're basically a piece of nucleic acid, a piece of DNA, and wrapped in a protein coat. That is also how they are identified for the most part, because the tests that we use to test for viruses is testing for that specific protein coat around the virus, because every virus has a specific protein coat. Viruses disrupt the cell metabolism, but they are also an obligate parasite. So similar to powdery mildew, they cannot survive outside of the host. Although powdery mildew can survive outside of the host, but viruses have that need to have that living host, and they do not have any survival structures that will allow them to survive outside. So they have to be in the plant material itself. And they are systemic, so they will go through the plant. And that's one of the things that when we talk about, well, how do you manage a virus problem? It's usually just pull the plant out, because it is systemic. There's no chemical cure, and you don't want it left in your garden, because insects could feed on it. And if it's an insect vectored virus, they could actually move that virus to other plants in your garden, and you really don't want that plant that they were discovered on. So for example, tomato spotted wilt virus was discovered on tomato, characterized, but that's not the only host. Tomato spotted wilt virus has over 300 host plants. So it's one of those things where, yeah, it's named tomato spotted wilt, but it could go to ornamentals, it could go to vet, besides tomato. So again, just knowing that can help can help you kind of understand the process of viruses. So one virus can infect one or dozens of different species of plants, just like I mentioned with tomato spotted wilt virus. Tobacco mosaic virus also has several hundred host plants. And even though tobacco was the first host plant it was characterized in, it can also go to tomatoes, potatoes, peppers, any of the solanaceous crops. It can go everywhere. So again, and it could be that, each species of plant can be attacked by a couple or several different kinds of viruses. And so sometimes you may have plants that are co-infected, meaning that they have more than one virus associated with those symptoms. Viruses do not have reproductive structures, nor do they have any overwintering structures. So they don't produce spores, they don't produce anything like Cleistothecia. And so consequently, they have to live in that host plant material. Viruses get moved around oftentimes by insects. Mechanical tools can move viruses as well. We can move viruses as, we touch a plant that might be infected with tobacco mosaic virus and touch a plant that's not infected. And just that, small interaction can move tobacco mosaic virus into that other plant. So we do have to be careful. Not every insect will spread a virus. The most common are going to be thrips, aphids, and there's some leafhoppers that we usually associate with virus transmission. Here in Colorado, whiteflies are not considered a huge issue as far as virus transmission goes, but in the southeast part of the country, they can be. There are a couple of viruses in tropical areas that they can vector. Areophyte mites is an arthropod, but it can also vector some virus as well as, and so can, so how do viruses move? Again, when we talk about moving from plant to plant, it's vectoring. And so how we vector a virus could be insects, could be mechanical, could be physical transmission, like we touch the plant sort of thing and go from there. Insects are probably the most common. Nematodes can also move viruses or vector viruses, seed, pollen as well. I'm not going to talk about any of those specific types of vectoring because for the most part, they're very specific and usually associated with cropping situations or, other things that we're not going to find in the landscape or the vegetable garden. So what we mostly talk about are mechanical and insect for the most part. So vector means an insect or other means of transmitting the virus from plant to plant. We can characterize insect movement by three different types. We have non-persistent, meaning that the insect picks it up on its stylet as it's feeding on the plant, and then it can transmit it right away. And an example of that would be aphids. Semi-persistent would be something in an insect where it has to get into the foregut of the insect before it can be transmitted back out. So that's usually, they can transmit within an hour to a 24 hours sort of thing. Persistent transmission, this is like leafhoppers and thrips, where the insect picks it up. It has to go completely through their system before they can infect a plant with it. And so it could be anywhere from, a couple of days to a week before they can transmit the virus again. So in terms of managing the persistent leafhoppers is probably the most critical because you can actually manage those a little bit better before they can transmit the virus than with something like an aphid, which can transmit it almost immediately after it picks it up. So viruses cause modeling, like this modeling color, sort of a mosaic, crinkling of the leaves. They can cause curling of the leaves. They can cause stunting or leaf spots. They can do different things like that. So again, oh, okay. So we see a lot of symptoms associated with this. So we have, like on this particular plant here on the lower right side, we see leaf spots associated on the leaves. And so you may say, well, it looks like kind of like a fungal pathogen, right? But it may not be. It could be that it is a virus, but it looks different on this particular plant than, say, on a vegetable. And that happened to me one time when I was, in the diagnostic clinic. I got a Dicentra plant come in, and they wanted to know what was causing the brown spots all over the leaves. And I thought, well, it looks like a fungal or a bacterial pathogen because it was very distinct spots. And so I cultured it. I was looking for spores. I couldn't find anything. It wasn't doing anything. So I emailed one of the listservs that I was on and said, I have this plant and I don't know what's happening with it. Does anybody have any ideas what could be wrong with it? And somebody popped up and said, well, have you tested it for tomato spotted wilt virus? And, this was a good lesson for me because I learned early on in my career that I can't always depend on the symptoms to tell me what's wrong with the plant. Because to me, the symptoms on that Dicentra looked fungal or bacterial. And then when he said that, I was like, well, okay. And then I tested it and it was positive for tomato spotted wilt virus. So it was a good lesson for me to learn that same virus, different host, different symptoms. So for example, on tomato, you might see something that looks like this on these leaves on the left, but this could be tomato spotted wilt virus. But the picture on the right here, this lower right, could be tomato spotted wilt virus as well. So again, all of these things combined together. And I'm not saying that plant pathology is easy, because it's not. It's one of those things that it takes a lot of practice to work with and to try and figure out what's happening. So I encourage you, don't get frustrated, because it does take a little bit of time. Okay, so tomato spotted wilt virus does exactly like it says. It causes spots and it causes wilting of the plant. So you can see the spots associated with here on the leaves, and then you can see the yellow spots on the tomatoes. Here they are on the tomatoes as well. So you can see all of these different associations. So I misspoke earlier. I said 300 plus hosts. Well, it's 500 plus for tomato spotted wilt virus is their host range. And it does affect the plant health, because it's not going to thrive as well as a healthy tomato plant. Now, you might ask, could you still eat the fruit off of this plant? And the answer is yes. These viruses will not affect humans, so you would not get infected if you ate this particular tomato. It's a little bit unsightly. It may not have the flavor that you're looking for. So again, trying to figure it out. I use the I use these tomatoes to make marinara sauce. So it's okay. And it's, to me, it works out well because, by the time you add all the herbs and everything in there and all the spices, it tastes pretty good. So for me to get tomato spotted wilt virus in my tomatoes, it doesn't matter so much. So how we identify this is with a test called an ELISA. ELISA stands for enzyme-linked immunosorbent assay. And in this case, tomato spotted wilt virus actually has one that's called an immunostrip. And basically what you do is you grind up some plant material in a specific buffer, and then you put this little stick in there and you wait 5 minutes and you get a result. What I tell my students is it's like a pregnancy test strip for plants. And so 2 stripes and it's positive, and 1 stripe and it's negative. So it's easy to tell. This is and other tests that are not quite as fast or quite as easy, but they work just as well. But it depends on what they're tested for. A lot of times with, we abbreviate this TSWV, you will see some purpling of the leaves, oftentimes some curling associated with it. You might see some necrosis, as in this upper right picture. Now, and these are on tomatoes. The lower right picture is on peppers, and the leaves in here. So again, you know, it's looking at this whole situation and trying to figure it out. And again, it's just asking questions. And oftentimes you'll find that this is a process of elimination. And okay, so we see this type of symptom. Do we find any thrips on the plant? Well, if we see thrips, because they transmit tomato spotted wilt virus, it's entirely possible that, yeah, this could be tomato spotted wilt. If we don't find thrips, well, the thrips could have left the building, but maybe it's a phosphorus deficiency, because that can cause reddish discoloration on the plants as well. And so again, you know, really it's a process of elimination. And this is a lot of information that I'm going over today, but it is being recorded. So you will be able to go back and look at this and, you know, kind of review pieces and parts that you have questions on. Tomato spotted wilt virus symptoms. Again, this is a fairly large tomato leaf. You can see the purpling over here in this picture on the right. You can see all of this purpling and kind of cupping, curling under of the leaves. This one picture on the left, you can see this associated with tomato spotted wilt. You've got all of these purpling colors and things like that. So again, you know, it just takes looking at these and asking questions and figuring out what's happening with the plant. So again, we have that wilting and spotting and crinkling of the leaf tissue. We can see ring spots on some plants. This is a begonia in this picture on the left that has the ring spots. So, okay, kind of matches the name tomato spotted wilt, right? So we get spots. Not every plant does that. Because again, remember, the virus is named for the first host plant it was on. And so not every plant will form these rings or spots. You might get cankers to form sometimes. It just depends, again, on the host plant and the virus. This is what the fruit can look like. And again, you can see in the fruit, excuse me. Okay, sorry. You can see the ring spots here on the tomato, and you can see here we have on this tomato ring spots. So you can see that discoloration, that yellow and red of the tomato. So again, you know, lots of different symptoms associated with this. And that being said, tobacco mosaic virus can look very similar to this as well. So this is why it's really kind of a challenge sometimes to try and figure out what's going on with our plants, right? And why plant pathology can be, you know, one of those things that just takes a little bit of time. So the flower thrips, Western flower thrips, or let's see, Frankliniella occidentalis is the name of the thrips that are the vector for tomato spotted wilt virus. They're very tiny. You do need a good hand lens pretty much to see them. They they don't technically have a pupil-like state. These insects are technically simple metamorphosis, where they go from egg, 4 stages of larva or instars, and then they become an adult. And so they don't technically have a pupal stage, but they just kind of molt into the adult stage. And they actually live in crevices on plants, or they live in the flowers. You'll often find them, if you're looking for them on plants, you will often find them right along the veins of the plants, because there's a lot of nutrients and a lot of food source in there, and that's where they like to feed. One of the characteristics of thrips feeding on plants is that it's kind of a silvery color, and oftentimes you'll see some blackish dots or spots inside that silvery color. I don't know if, yeah, I do have a good picture of it here. So this is thrips injury. This is what we talk about with the silvery color on the plant material. This is where the thrips have been feeding. And you can see it's right along this vein area where they're getting a lot of nutrients. These dark areas right here, this is their first, and this is a polite way of saying insect poop. So this is their insect poop, and it's this really dark greenish color. So it looks black, but it's really a dark green because basically what they're excreting is the chlorophyll that they've taken in from feeding on the plant cells. So you can see all of this right here. And so you can see this very characteristic of thrips feeding on the plant. If you see this, you've gotten, or you have had, or you do have thrips. Now, they also like to hide inside flowers, and they will feed on flower petals. So you'll see this flower scarring associated with that. And then if you look on the upper side of the leaf, again, you see this silvery type pattern to the thrips injury. So it's pretty distinctive. And this is the way it looks on almost every plant. So not just tomatoes, but they vectored tomato spotted wilt virus to a lot of different host plants. And so you'll see this silvery type feeding damage on a lot of different host plants. So you would be able to recognize that. So how do we manage this? Well, we can use spinosad, which is an insecticide. That's very good for it. Oops, wrong way. And there are some resistant varieties of tomatoes, depending on what you like to grow. I've heard that some of them don't have very good flavor, but I don't know for sure. I've never tried to grow them. Get your infested plants out of there and destroy them. I would not put these in a compost pile, because if the plant material does not break down fast enough, the virus can still survive in undecomposed plant material, and the thrips fly. And they can still feed on that undecomposed plant material, pick up the virus and move it to another plant. They can vector it into another plant. So I really highly recommend just disposing of these, putting them in the trash, covering them up, sealing them. Really important to make sure that we're managing that correctly. Now, you might also ask if viruses can live in the soil, and the answer to that is no. The only way viruses survive plant, if you have undecomposed plant material in the soil, then potentially the virus could be in that undecomposed plant material. Viruses, by their very nature, need a live host to survive in. So they will not survive in the soil. They have nothing to feed on. They have nothing to live in. There's no cells for them. There's no nucleus, okay, in the soil. So they have to have a living plant host. That being said, undecomposed plant material can still harbor the virus if it is not completely decomposed. So if somebody tells you that, well, I have a virus in my soil, you can confidently look at them and say, no, you do not. You may have plants in your soil that have the virus in it, but soil cannot support a virus. So you can definitely say that. So again, managing thrips can also be an issue, making sure that you have, don't bring plants in that have thrips on them, or, just keep an eye out for those feeding symptoms so that you can use an insecticide. You could also use an insecticidal soap. If you problem with thrips is they're tiny, and the insecticidal soap is a contact, and so you really have to get it on top of that thrips in order for it to kill the thrips. Some other virus examples that we see, like rose mosaic virus. I showed this one at the beginning this morning, and so we could see this. We also have peony ring spot virus over here, and I've seen that a couple of times. I don't know as there is a good test for that one, but again, the symptoms are pretty distinctive. For mechanical transmission of viruses, tobacco mosaic is the classic example. And this virus affects a lot of other solanaceous hosts. So we have, 100 plus, 200 plus host plants for tobacco mosaic virus. Now, this is tobacco picture is infected with tobacco mosaic virus. And you can see the light and dark colors here, as well as this kind of puckering of the leaves. And this puckering of the leaves, again, this is where the plant pathologist comes in. This is called rugosity. And this is associated with tobacco mosaic virus. And it's the term that plant pathologists use for this particular symptom. Instead of just calling it puckering, we call it rugosity. So tobacco mosaic virus is mechanically transmitted. So that means we can do it. It can also be moved by pruning materials, your pruning equipment, things like that. And now we have, again, there's resistant varieties. It causes fruit distortion. You can see this, but you can also see the ring spots, as in this top tomato here. You can see these kind of like scarring across the tomato surface. You can get some leaf yellowing in the bottom right picture here with the tomato leaves. You can see the yellowing versus the dark green color of the tomato and tomato leaves. And so you would be able to, see that as well. Here on this pepper in the left-hand picture, you can see the rugosity, this puckering, as well as the light versus dark coloring of the leaves. So you would be able to see all of this associated with that. And now the reason I say that we can transmit this virus is because even if we, touch a plant that has tobacco mosaic virus and we don't think we've done anything, all it takes is one cell to break and it's on your fingers and you don't know it, you can't see it, but you can touch another plant, break a cell, an epidermal cell, and that virus is transmitted just like that. So it's one of those things where we have to be very careful. So wearing gloves or changing gloves between plants can be helpful. Pruning equipment, washing our hands on a regular basis is really important as well for reducing the transmission of tobacco mosaic virus. So phytoplasma characteristics. Let's talk about phytoplasmas. Phytoplasmas are basically bacteria without a cell wall. They do not have a cell wall. Consequently, they cannot be cultured in artificial media. And it's very difficult to do, if not impossible, to do transmission research with phytoplasmas, because even if you were to purify it in a plant, pull it out, trying to inject another plant with that phytoplasma is going to be a little difficult. And again, you can't see the cells under the microscope either. These are also obligate parasites. They do need a live host plant in order to survive. So again, one of those, obligate parasite plants. So once the plant dies, those are often vectored by leaf hoppers. So for example, let's see, what is a good example of a phytoplasma? In carrots, aster yellows and carrots, we see some purplish foliage as well as really hairy carrots. So the roots of the carrot are really rooty, have a lot of root hairs on them. And this is moved by the aster leafhopper. So they pick it up in a plant. And since this is a persistent vector, it can survive in that leafhopper for, the life of the insect, and they can vector it every time they go feed on a plant. They can move that. So again, it's, one of those things where trying to catch the vector is going to be a challenge, because leafhoppers, they're, very cyclical. We don't always see them. They can just move in and out, and sometimes they're here and gone before we even know that they've done the damage. So looking at those. Other symptoms caused by phytoplasmas that we often see are things like sterile flowers, like this picture in the bottom on this echinacea, where they're not coloring up, and they're also immature. They will never produce a true seed. They're sterile. You might see yellowing or purpling of the foliage. You might see something like these rat tails appearance. And sometimes there's distortion associated with those as well. So some of the common phytoplasmas that we see here are ash yellows and aster yellows, and that's like carrots. Sometimes we could see it on ash trees as well. But it's very difficult to diagnose these. It's a molecular test for phytoplasmas. There's no ELISA. You can't culture them, can't see them with a microscope. So really the only way to identify these is, again, with molecular testing. So how do we manage these things? Well, pretty much it's managing the insects for insect control, so that the leafhoppers don't transmit the disease or the pathogen. We can take out the infected plants so that if there are leafhoppers around, they can't feed on the infected plants, and then move it into uninfected plants. So again, looking at these sorts of things, if you have, plants that are susceptible, you have chemical controls, you have repeated sprays, things like that, those can all help for managing. So again, we have our leafhopper. Okay, parasitic plants. This picture is an example of dwarf mistletoe here in Colorado. So dwarf mistletoe is Arsuthobium, and I can't remember the species now. It's an Arsuthobium genus, and these are the dwarf mistletoes. So these are not mistletoe sort of thing. That's a true mistletoe, and that's considered a leafy mistletoe. And those are usually phorandendron species. I don't remember off the top of my head, but we don't have very many of those in Colorado, if any. What we do have are these dwarf mistletoes, but like I said, these are going to be in the foothills or the mountain areas. The entire life cycle of dwarf mistletoe can take anywhere from 5 to 8 years, depending on environment and how fast it spreads, drought, different things like that. And what happens is the mistletoe at the end of its life cycle will produce a flower. And these flowers will eject a seed that can land on another host plant. And that seed is covered with this sticky mucilaginous sort of matrix that allows it to stay where it lands, so it won't roll off the tree branch sort of thing. And so the first symptoms that you're going to see are something like this swelling of the branch tissue. And, it might just be a slight swelling that you don't even notice. And then a couple of years later, you're going to start seeing these protrusions coming out of the branches. And they're usually yellowish colored because they don't produce their own food. They are feeding off of the plant itself. That's why they're called a parasite. And so they don't have any chlorophyll. They don't photosynthesize. They are feeding out of the vascular system of the plant. And so you start seeing these yellow protrusions, and then as the mistletoe ages, it becomes bigger and bigger, and it kind of it can produce witches brooming in the trees. It can produce these bright yellow colors, and sometimes they're very distinctive. You can actually see them almost as you're driving by. And so it's one of those things where it's really, really distinctive and noticeable. They reproduce by true seeds, like I said, and even animals, birds and things like that can pick up these seeds if they're crawling in the trees, and they can move seeds from tree to tree. So that can happen as well. So again, they are a parasite, and that's how they damage the plant. They are taking nutrients from the plant to sustain the mistletoe. And so consequently, they don't outright kill the tree, but they do become stressed and the trees will eventually die. And how do we manage this? Generally, you can do it by pruning, but oftentimes if you are living in the mountain property or your clients have property up there, realistically, how many trees are you going to be able to prune? And mistletoes, these mistletoes are host specific. So for example, ponderosa, dwarf mistletoe will not go to lodgepole. Lodgepole will not go to ponderosa. You might be able to different tree species that are not susceptible to the mistletoe that you have in your area. So for example, if you have a lot of ponderosa mistletoe, maybe plant some Douglas fir because that's not susceptible to ponderosa mistletoe either. So again, you know, talking about all of these things and seeing what's happening can be, you know, just looking at these things and trying to identify them can be important. Nematodes are considered microscopic roundworms. They are not true worms. You would not be able to see a nematode ever without a microscope. So if somebody comes to you and says, "Ooh, I found these white things," and they're holding them in your hand and you can see them with the naked eye, those are going to be true worms. Those are not nematodes. You would not be able to see nematodes with the naked eye. You have to have a hand lens or a microscope in order to see these. So for example, you know, when I'm digging around in the soil and I'm working the soil, I'll often see these little, you know, wormy things. And it's like, oh yeah, no nematodes there. Those are worms. Plant pathogenic nematodes have a structure in their mouth that's called a stylet. And it is basically how they feed on plants. So they eject that stylet to pierce the plant itself. And so they can feed on the cell contents. They do need a little bit of free water because they do move through the soil in water. Now on plant material itself, because we have different kinds of nematodes, we have foliar feeding nematodes like what is it? Aphelincoides are the foliar nematodes, I believe. We have soil-borne nematodes. So a lot of those are the cyst nematodes like Heterodera or Meloidogyne that cause cysts on roots of plants. We can also have stem nematodes. These are things like pine wilt nematode is actually considered a stem nematode because it affects the trunk, the main stem of the plant. It goes into the stems, the branches, and the main trunk. So that would be considered a stem nematode. And then we have bulb nematodes. And those would be things like that get into onions and garlics and those sorts of things. So again, you know, different, we also have foliar nematodes or seed head nematodes. So things like anguina nematodes that take the place of seed in turf grass and other grains. So those are some of the things that we look at. They do reproduce by eggs. So we have eggs, and then there's 5 juvenile stages before the adult. They do require free water. Like I said, in the soil, they can move through films of water in the soil. They don't move very far. They don't move very fast, but they can move through the water. And so those are some characteristics of nematodes. Now, again, you can't see these with the naked eye. You do need a microscope. It's always best to have these identified. So, you know, if you suspect you have a nematode problem. This is an example. This is what the stylet looks like. This is what the nematode will push out and into the plant. It will inject the plant with this stylet. This is the mouth. This is an electron micrograph of the mouth of a nematode. So you can see it's right here. And that's where the stylet would come out of. And remember, only plant pathogenic nematodes have stylets. So not, and you know, and we do have good nematodes that are in our soil, they decompose organic matter. They're just kind of hanging out. They're not really doing anything. So it's really important to make sure that if you see, if you think you have nematodes in your soil, make sure that they're plant pathogenic, because we do have beneficial nematodes as well. So nematodes, again, this is what a cyst nematode damage would look like in this left picture, upper left picture. The bottom picture here is a foliar type nematode. So this would be, I believe, the Aphelincoides nematode that does this type of damage on plants, on foliage, on leaf material. And then this upper right depiction up here is the pine wilt nematode. This is Bursaphilincus xylophilus, and this is vectored by the pine sawyer beetle, moves this particular nematode around. And we do have pine wilt nematode here in Colorado. And Dr. Stewart is also working on this for, as part of her program. She's working on pine wilt nematode, and has now confirmed that here up in the mountains where I live in, up by Red Feather Lakes, that we do have pine beetle in our area. So it is moving. Not as fast as some people think it is, but it is moving. And so we do have to be cautious with this and watch for it. Some of the symptoms of nematodes, again, like I said, are those cysts on the roots. We have these spots or these discoloration of the leaves. Could also be stunting, yellowing of the plant material, wilting, eventual death of the plant. And like I said, nematodes can move around. Insects can vector those, like the pine sawyer vectors the pine wilt nematode. It can move in soil, infected plant material unknowingly brought into an area. They can move in water, but not very far. They're not long distance swimmers, but they can be spread in water as well. So how do we sort this all out, right? When's our next break, Cassie?
I was just about to interrupt you and say it is now.
Okay, cool. Well, let's just take what we have, 15 minutes, and
10 minutes.
What, go till 10. Okay. So
And when we come back, we've got a couple questions for you through the Q&A as well.
Absolutely. Got it. Okay.
We do have a couple questions in the Q&A that we wanted to make sure that you were able to answer for us, Tamla. The first one was actually from just before lunch about whether or not you could talk about fungal issues on seedlings, and how to diagnose common fungal issues on seedlings, like when you're starting seeds.
Well, it would be very similar to identifying root problems. The thing of it is there's several different fungi that can cause root problems on seedlings, and also it depends. Often, if you're using a potting mix that doesn't have any native soil in it, then chances of having root rot are slim. Simply because there's no native soils. And in our native soils, we can have like pathogens that are just hanging out until something is planted in them, and then they can start causing problems. So it depends. I mean, we have things like Rhizoctonia, Pythium, Fusarium, Verticillium. Let's see what else. Those are the most common ones that we see as far as causing damping off of seedlings. And if you're not using native soil, then it shouldn't be too bad. There are some, I don't know if there are any, I was going to say, I don't know if there's any soil drenches that can help with that. However, it would be, you could check into it. You could also pasteurize your soil if you're using true soil or native soils, you could do that. But again, it would require either culturing or some sort of microscopic identification of the particular pathogen to see which one it is in order to identify it.
Great, thank you. And our next question is about dwarf mistletoe, a little bit longer. If you remove the mistletoe from the tree limb, will more come out from the limb? There are some areas around me with ponderosa dwarf mistletoe. Do you know if the Forest Service will respond to reports and prune trees in the area, or is it a lost cause?
Good question. Okay, so dwarf mistletoe is kind of like what I was describing with fire blight, because the mistletoe can be growing through the branch. You just don't see it projecting out of the branches yet. So even if you take off the mistletoe that you see, you're still going to have some mistletoe coming out of the branches. And because it could be moving back and forth in the branches, things like that. And so you would have to prune off, say, the entire branch, maybe, and that might get rid of it. That being said, it's a little difficult to do that a lot. And another thing is I don't think the Forest Service really cares about dwarf mistletoe. I don't think that if you called them, I don't think that they would do anything. Because in a native forest area, it's kind of like, well, Mother Nature sort of takes care of itself, because as trees are declining, there's new seedlings that are coming up naturally. And so when those other trees with the mistletoe decline, then hopefully the other trees will be large enough and, provide some sort of forested area. Um, uh, you know, where I live, of course, we had Cameron Peak fire last year. So I'm sure that destroyed a lot of mistletoe. But uh, uh, as for just coming up and pruning it out, I don't believe the Forest Service would do that. So, yeah, I'm sorry. I don't have better news for you on that one.
And that's all I've got at the moment. Is there anything you've got, Mari?
Okay.
I wanted to see if Tamla could clarify the virus concept. There is some controversy that they are alive or not alive. I just wanted to hear your thoughts on that.
Oh, okay. Whether viruses are alive.
Mm-hmm. Yes.
So viruses do live in the plants, and in that sense, they are alive. But once the plant dies, the virus dies because it doesn't have a plant to live in. So whether a virus is truly alive or not is kind of subjective. If it's in the plant, it's alive because it's feeding on the plant, it's replicating in the plant cells. If the plant has died, the virus has died, so it's not going to replicate, it's not alive anymore. Does that help?
Yes, I think so. There was just some confusion that because they need a host, then they are not alive. But it is the same concept as if you were the powder mildew, it also need a host, but it's alive. So it's kind of like that same concept. Yeah, thank you.
Yeah. And it is kind of confusing because the viruses can survive in the plant and they're alive while they are in the plant, but they don't survive without the plant. So in that sense, it's they're dependent on the host plant in order to survive and to live. If they don't have a host plant, they don't live. They can't do that. So you can do that. Mm-hmm.
Great, thank you.
Okay, sure. All right, is there anything else before I share my screen again and we keep moving forward?
I think we can move along.
Okay, so we're going to be wrapping it up here shortly. You guys will probably be done. I hate to say this, but we're going to be done by 2, I'm sure, unless we have a lot of other questions. But because this is kind of the wrap up, putting everything in perspective, we're talking about how do you sort it all out, right? You know, and this is one of the challenges when we start looking at plant pathology is how do we figure all this out? And some of the generalities that we can talk about is with abiotic issues, it's generally going to be uniform. You won't see any signs. There's not a sign of a plant pathogen or anything like that. And you can see it over numerous species of plants or different types of plants. It's not necessarily going to be just one. If it's a biotic issue, meaning it's a pathogen or something else, generally is going to be more random. We may see signs of a pathogen, so either the spores or like powdery mildew or oozing or things like that. And it's usually confined to one species or plants in the same family with related species. So you can kind of, start eliminating based on that as whether you're looking at an abiotic versus a biotic issue. So, we look again at random versus uniform. So in this example, on the lower left picture, we have our African violet, and it has a few leaves that have some random damage. But up here in this upper right-hand picture, we see very uniform damage. So you can look at all of these and you could say, these all got planted, looks like they all died, it looks like. So now we got to figure out what abiotic issue were we dealing with in this particular area. Did the water get shut off? Nobody watered them after they planted them? What was happening with that? So we can start looking at, random versus uniform. Okay, so now we look at how these symptoms and signs progress. We're looking at how they progress. When we talk about an abiotic problem, generally it's a one and done sort of thing, no symptom progression. The only exception to that, I shouldn't say the only, but the common exception to this is fertilizer or nutrition issues with plants, because those can progress slowly. They can move throughout the plant as the plant becomes depleted of whatever nutrient it needs. The symptoms can become more severe. With a biotic problem, the symptoms progress, and sometimes your nearby plants can become infected, especially if they're in the same species or the same genus sort of thing. So we have to be careful with that. So we look at all of those things associated with that. So a biotic is going to progress, abiotic generally not going to progress and get any worse. We also look at cultural practices. So, when we try and figure out what's going on with our plants and we're asking questions of our clients as to how they're taking care of plants, these can also help figure out whether we're looking at an abiotic issue or a biotic issue. So for example, when was the plant planted? How was it planted? Did they use proper planting techniques for the type of plant that they're putting in? Have they used fertilizers? Any chemical applications? Could it be phytotoxicity based on a chemical? Have they used any insecticides or herbicides or fungicides? How often and how much do they irrigate? Can also be a good question. Are they hand watering or using an irrigation system? I know I've been on a few lawn calls, lawn checks. Well, not lately because of COVID, but going on lawn checks, and a lot of times, from what I've heard, that like 90 to 95% of the problems are an irrigation issue. The heads are broken, the clocks haven't been reset. There's not enough coverage for the area. There's not enough zones for the area that it's trying to water. All sorts of different things can happen with improper irrigation. And so looking at whether it's hand watering, how much water is put down, that can all play a role in trying to figure out what's going on with our plants. Soil type, what is there any compaction, anything going on like that? And some of these things we look at, like if turf grass problems, if they have a large thatch layer, like over half an inch, chances are that's going to be a problem. And you can tell that this, I don't even want to call this soil down here underneath this thatch layer, but it looks like clay and rock, and it looks very dried out. And so like it's not getting enough water, probably not being core aerated, all of these things associated with it. And so chances are they're probably going to have an issue. A lot of times what we see with our plants is that oftentimes it starts as a cultural problem or an abiotic issue, and it doesn't get fixed. And so that cultural issue, that abiotic issue, leads to stress on the plants, and then they become more susceptible to pathogens, and that can cause problems with that particular plant. So again, looking at all of these things, and then when we talk to our clients and they're like, well, I have this disease, but the disease may be secondary, and whatever was stressing the plant is the primary problem. And then the disease just took over because, hey, it's, got an easy entry into a stressed plant. And so that can be an issue associated with that as well. We also want to look at symptoms. And I know that you probably saw some of this last week with the IPM and plant diagnostics, but it's always good to, pay attention. When we're looking at symptoms, we're looking at patterns. And we look to see what's happening. So for example, in this picture, we're looking at, looks like tire tracks driving across a turf situation. So was a turf frosted? Did it have a pathogen on it? And as the equipment moved across the turf, it spread the pathogen. That's a possibility. Is the turf droughty? Does it need water? Is that why it's marking up there? Was the turf covered in frost when it got driven across? Those are all things to look at and to ask as well. The hostas in the bottom. So you have several different varieties of hosta in here, and they all have these same similar shredded leaf symptoms. So to me, this looks like hail damage. Hail damage that, happened, maybe a summertime storm came through, but they all have the same symptom. Now, that being said, if we haven't had any hail, I'd probably start looking for snails, slugs, because they like hostas. And so then I'd start looking for those. And they come out early in the morning or, before dawn. So you'd have to get up a little bit early to see that. But, it could be a couple of different things looking at those. And so making sure, checking out what's happening. We can look at patterns again, and looking at all of these things, pattern distribution. Conifers actually have what's considered to be a circular vascular system. So it kind of looks like a stair step going up the tree. This picture on the left, you can see it's kind of at an angle on the bottom, and then it's an angle in the middle, and then it goes up to the top. And the same one with the picture on the right. You have this kind of angle system here. So what this tells me is that there was some damage to part of the root system that affected this particular area of the vascular system of the tree. So you can see things like that. And if you've ever seen standing dead trees up in the mountains, you can see this circular system of a standing dead pine. And you can see how it would be kind of, it's good to kind of have that whole picture and see what's going on with the plant. So you would be able to see that. Here we have some abiotic problems. You can see it's kind of a random sort of going through here. This could also be, it looks like it's kind of a stair-stepping thing on the plant as well. So it might be the might be a root system problem, that maybe a root got severed, somebody put in an irrigation system, or they were doing some construction or something like that. So there's all sorts of things to think about when we start seeing things like this. This particular plant, this tree, they probably should have just taken that out. There's a lot of construction here. They're doing work. Looks like this is a concrete with brick. And so they've turned the water off, but you're getting a lot of heat coming off of there, and you're just stressing out this tree. So it probably would have been a better idea just to take that thing out and put something in when you're done with the construction, because there's probably no way that this tree is going to recover. This is almost 50%. We're looking at least 33 to 40% of this tree is affected like this. And that's not good if you have that much of the tree that's looking like that. When we look at our plants also, we want to look at growth increments. This is probably the easiest way to tell how a plant is growing. And it's a good overall health clue for the plant itself. So we're looking at these. This is on a deciduous tree, and you can see there's a growth increment here, growth increment here. There's another one here, and then we have one here. Well, this is from here to here, could be about maybe an inch, a little bit more. From here to here, it's about the same distance. But from here to here, whoa, what happened? So, well, that tells us, okay, something happened in the last year, because this is current year growth. This is previous year right here where this blue arrow is. And then we kind of move back along the branch, and we can see, so we're going back 4 years already. And we can see that something happened in the previous year. And so then we can ask, okay, what happened? Did the water get turned off? Did something happen like that? Different things like that. And it's easier to tell on conifers for me anyway, to look at those overall growth increments and see what's going on. So our final steps in a plant problem diagnosis is not only are we looking at symptoms and signs, determining whether it's abiotic versus biotic, looking at environmental conditions, looking at how the symptoms and signs are progressing, reviewing cultural practices, observing patterns. We also want to ask questions, and then we consult our literature and our control measures. When you're looking up information on the internet, sometimes when we're doing that, the internet can be our best friend, or it can be our worst enemy. And one of the ways to limit some of the information that you're getting is to use site:/edu after your search term. That will limit your results to hopefully scientific-based, peer-reviewed, research-based information. And so you can be, fairly confident that you're using something like that that can review that. Not always is everything filtered out that way, but for the most part it is. And so you would be able to be confident that what you're looking at is research based. And you can use other land grant universities. You can use Colorado State University. Utah State has some great information, especially for the Western slope, because their conditions are similar. Topographical and environmental conditions are similar. So it really is, a good way to do that. And when we ask questions, we have to consider that a lot of people may not know all of the information that you're asking. And sometimes we just have to keep a question at a time and figure it out. There can be, there's about 6 major symptoms for hundreds of causes, wilting, yellowing, things like that. But it could be anything from abiotic to, viruses to other plant pathogens and things. And oftentimes it's the primary cause is an abiotic or cultural issue, and then it becomes a pathogen issue because the plant is stressed. And so again, looking at all of these things can be really important, just to ask questions and figure out what's going on. So, the more questions you ask, the more clues you get. Remember, we're playing Sherlock Holmes here. We're being detectives, trying to figure out what's going on. Where is the plant in the landscape? How is it exposed to northern winds, southern light? Is it, where is it located? When did you first notice what was going on with the plant? And I can tell you from experience that most people will say, well, it just happened. And I didn't see anything. And because a lot of times what we're looking at doesn't progress, or it's so slow progressing that people don't notice it. And so consequently, we have to just keep asking questions and things like that. We look at the time of the year that we're looking at these plant samples. What kind of weather concerns have we had? And, for me, it's like, well, now we have a snow up here, which is great, but it's going to be one of those, well, how are we ever going to get out? Right. And so I'm really, I'm glad, because it's fire insurance for us. But, for other people, the snow might damage plants. Up here, in the mountains, the plants are kind of used to this. And so we don't have the plant damage that we associate with down in the foothills, or down along the Front Range. How has the plant been cared for? Has it been watered on a regular basis? Do they fertilize? Have they treated it with anything? Do they take care of the landscape themselves, or do they have a service do it? And do they have records if a service comes and takes care of their landscape, so that they know? So again, just asking lots and lots of questions to help us get to our answers. We can look at branches, we can look at leaves, and try and determine it. This up here it could be a leaf scorch, but because it's also down here, I would go more with pathogen on this one, and maybe think more along the lines of anthracnose. This one to me is consistent with a dicamba issue, a herbicide issue. This one looks like tar spot on maples. Okay, so you have lots of different things that you can look at and you can look up. And like I said, Whitney's book is probably your best overall reference. It may not have everything in it, but it's probably one of the better ones that's the best one for our area. And so that is also a good thing to have in your library. We look at the flowers. We can see, okay, what kind of damage is on the flowers. We can look at the leaves and see what's happening there. I mean, is it insect? Is it fungal problems? What are we looking at? We can look at things, and a lot of times we're not going to be able to see like lower trunk or upper trunk. It depends on the county that you're in and what's happening, because what's going on is maybe your county's not allowing master gardeners to go to homes yet, or you're not allowed to get out there to look at these things because of COVID and the pandemic response. So it just depends. You do have to follow your county regulations if you're going into the master gardener program and looking at that. So your best bet might be to ask for pictures. And, a lot of times what I like to ask for are pictures close up of the problem, and then the macro view, back off and take a big picture so I can have some reference as to what I'm looking at. Because sometimes I'll just get a picture of a close up and I have no reference as to where this is on the plant, what they're doing, anything like that. And I may not even be able to tell what kind of plant it is. If I just get a picture of bark, that's kind of a challenge. So we may not be able to do that. So some of the things that we can look for also, look up our references, read it, is there a lab test needed? Where do you go? CSU has the diagnostic clinic on campus at CSU right now. and it will be moving down to the Spur campus in Denver, probably in the next couple of years, as soon as they finish that. So it won't be up on campus anymore. It'll be more centrally located. There's also the Jefferson County Diagnostic Clinic that can do diagnostics out there. So, you know, that's a couple options for you to look at. Do you need more information? Make sure that you have contact for your clients so that you can ask them questions and see what's happening so that you can do that. And then, you know, analyze the information. As master gardeners, and I know that not all of you are master gardeners, but as master gardeners, our job is to give out research-based information, and we give our clients all of the information and let our clients make the decision. I didn't say this earlier, but I started my career as a master gardener back in the 90s. I was a master gardener in Adams County. And this was one of the things that was always drummed into my head, was that it is scientific, it's research-based information. We give the clients everything they need to know, and that client will make the decision. We do not, we park our personal stuff at the door. And so we're not allowed to do that. We just don't do that. I'm sorry, I'm laughing because Cassie just messaged and says she still has my file in Adams County. How funny. So do you need more info? You know, analyze the information. So we have to be objective, and we have to do this because this is what CSU does. This is what Extension does, is we provide that research-based information. So, so we start with biotic. At least when I diagnose, that's what I like to do. I start with the biotic issues. So I'm looking for insects, looking for pathogens. If I don't find any of those, then I move to abiotic. Okay, what potentially could culturally be happening to this plant? And I'm looking at primary versus secondary factors. And remember, I said primary factors oftentimes are going to be cultural, and then contributing factors are going to be those pathogens that come in because our plants are stressed. So again, it's one of those things where it's like, yeah, you know, we have to look at everything when we give our answer. So what's really causing the problem versus what is contributing to the problem. So this poor plant here, the primary problem is it's the wrong plant in the wrong place. It's in a parking lot. It's got weeds all over the place, probably not getting much water, and they want it to grow. It's got this little plastic thing around the base to keep the rabbits from feeding on it, but it's stressed out. I mean, that's just doesn't have a whole lot of leaves on it. So it could be getting cytospora canker, which is a contributing problem. And so people will say, well, it has a canker, and so that's the problem. And no, not necessarily. So again, it's one of those things where we have to look at it. Primary versus secondary. So when we look at our findings and we're looking at this, is it a problem or is it normal? Is it affecting plant health or will it? Is it affecting aesthetics? We also have to think about, what does the client want? What can reasonably be done? And then we give them all the research-based information and let the client decide the course of action. And I say that mostly because not only is that what we need to do as master gardeners, but from a personal standpoint, I will have people call me and say, well, I have this problem with my plant. What would you do? And honestly, I cannot answer that question, because my idea of a landscape and aesthetic is going to be completely different than somebody else's. I live in the mountains. I don't really have a landscape. I have, a few perennials around the base of my deck, and I have my big garden that has raised beds in it. That's my landscape. Everything else is Mother Nature takes care of. And so consequently, I'm like, I can't answer that, because, well, would you cut down this tree? If it was me and it had cytospora canker, yeah, I'd cut it down, but I'm not going to tell somebody that, because that's their decision. and then if they come back and say, well, you told me to cut it down, then, then we got a problem, because then they get mad and they're upset. And so as master gardeners, we cannot answer that question. We just have to give them the information and let them decide what they want to do. Everybody's aesthetic is going to be different. There are some people who want that pristine lawn. They want the perfect perennial garden. There are other people who want a native landscape. They're planting native flowers all over the place. They just want it to be natural. There's, people who want in between of those two things. And so consequently, again, we give our clients the information and let them decide. So that's the best thing that we can do. All righty. That is the end of my presentation today. Do y'all have any questions?
Have you ever heard me beatbox?
We don't have anything hanging right now. Let's give everyone a few minutes and see if anything comes in the chat. But
Sure.
Otherwise, I want to say thank you very much, Tamla.
You're welcome.
Hey, Tamla.
I hope my file is decent.
Tamla, do you have a comment on like those apps that circle around about plant diseases and diagnostics? I don't particularly, but I don't know if you have anyone that you will use.
I don't for sure. There was one out of Purdue that was doing a plant disease, but it was, I hate to say this, but some of the apps are out there. You have to make sure that it's going to work for our area, because the further east you go, you're going to find different pathogens and different problems, simply because it's more precipitation. The further east you get more precipitation. And so it's one of those things where you have to just kind of look at what's happening. And I thought that, didn't CSU Extension put together one of those, or was it a plant ID one? I thought Mark Platten down in, what county is he in, was kind of instrumental in putting together an app.
I know there's a
for
Woody
CSU
Plant ID
Extension. I
app,
don't remember.
but I don't know of a disease
That
one.
might be it. Okay. Purdue. Yeah. Let me see. Let me just check my phone here while I'm talking to you, because it may be in the apps. I just don't use it very often. Oh yeah, Purdue Tree Doctor. Let me see. Okay, this is Purdue Tree Doctor, and you can look at, oh, okay. It's telling me the app was built for an older version of Android. So I'm sure I have to update it or something, but this one actually cost money. So you might not want it as a freebie, but it has some diseases in there, things like that. And if you touch on any one of them, you will be able to see pictures of symptoms of that. You can see close up. So like I just picked on one called Black Knot of cherry, and it actually looks pretty good. I don't think you guys can see this very well, but this is what it looks like. Let me see, it just disappeared. There we go. Okay, so this is it. Let me go back and see if I can show you yeah, it's called Purdue Tree Doctor. I don't know if you can see it very well. Nope, it just disappeared. There we go. Purdue Tree Doctor. And so you might look that one up. But again, they probably have stuff there that that we don't have here in Colorado. So you'd have to make sure that you're looking up that information as well to see if it actually occurs in Colorado. Does that help?
Yes, thank you. I actually Google it, and they also have like Purdue turf doctor, tomato doctor, flower doctor. So they have all those resources, but keep in mind that Purdue, or where Purdue is located, is very humid. So as you said, the diseases are going to be quite different.
Yes.
But
Yeah.
thank you. I think that's helpful.
Yeah. And so, sure. Yeah.
All right, and we had one other great question come in. If cultural practices are primary and pathology is secondary, why start the diagnostic process with biotic causes?
So the reason I do it that way is because cultural practices are primary, and the biotic causes are usually secondary. So, but I try to do that because probably 80% of the plant problems that you're ever going to come across are going to be biology or going to be abiotic. And so you have to look at that, and then we go to the biotic, because the biotic actually involves a lot more. Well, I don't want to say it quite like that, but again, it's I start with the abiotic because it is cultural, and it is the primary. It can be the primary cause, and so then the pathogens are secondary. So that's why I do the pathogens after the abiotic.
All right, I think most of the questions we've answered, and we've had a ton of people saying that this has been a great presentation. And thank you so very much.
Thank you. Good, good. I'm glad you all enjoyed it. It's been fun. I love my plant pathology. If you can't tell by my vocal intonation, I am quite excited about it. So, I do enjoy it.
And somebody just said thank you for your dog as well. They enjoyed hearing your dog.
Yes, sorry about that. Yes, they are a handful. This is true.
All right, well, thank you so much, Tamla. And if folks have questions, I think your email is on Canvas, correct, Tamla?
Oh, it should be, yeah.
Yeah, so you can email Tamla, or you can email your local coordinator or agent for clarification on questions.
Yeah, it's all good.
All right, thanks so much, everybody.
Excellent. Well,
we'll see you again next week.
Thank you all for your attention. Have a great day.
We'll see you all next week for vegetables.